Uh-huh.
on April 4th, actually. So that's very soon.
Hmm.
And then I'll put in the chat actually what I did find out.
Mm-hmm.
So they have a fair coming, but then at the same time, it's not registered as a chapter or organization.
Mm-hmm.
So I was thinking maybe if GMA has like a spot or they're gonna get a spot, they can promote it there like together.
Mm.
And then in the meantime, someone can submit, a Northwestern student can submit a proposal to make that a chapter, like
Mm.
registered within their school. Because I think having that exposure within like student organizations, if
Mm.
there's other health clubs there, can get like more members to work on tasks and also get the exposure that we need and meet its target audience. So the deadlines that are coming up for that would be like April 15th and April 4th. So
Mm.
I think that's something that Northwestern students can look into.
Yeah, that's a great idea. Can you talk to Sally? I think she can help out. And then I think yeah, Chloe is likely going to be the next president or something.
Mm-hmm.
How do you
So
Okay.
yeah, I think it's a good idea, and I do think we will
Can you send
register.
send it?
Mm-hmm.
Yeah, we'll register as a chapter, and I think Sally's also registering as like the like actual like US NGO first, and then make
Mm-hmm.
student chapter in Jima and hopefully in Feinberg as well, right, Stas? We'll do a
Well, it just,
chapter.
yeah, it really depends. You just need a group of students, M1s probably, who can put in the time and put it all together.
Yes, yes. So yeah, let's work on it. I think we need to do some recruiting to get this really going. So Shauna, yeah, please work with Sally and see if we can promote during that event and make sure the organization proposal is submitted. We can start with some of the GMA members
Mm-hmm.
who's interested first. And yeah, in the same token, I'll get back to you, Shauna. Stas, any
Okay.
so about registering in Feinberg chapter, what are the pending processes?
Let me send you a link. Let's see. I think hold on, give me one sec. I have the link saved to the requirements. They're extensive, but it's doable. You basically have to get a group of students together and fill out some paperwork and then present the paperwork to the student senate.
How many students are required?
I think 10,
Well, that's a lot.
but
10,
I'm not sure. Oh, here it is. Copy link.
10 Feinberg students.
So I just linked the guidelines in the chat.
Okay.
Yeah.
Okay. Eugene O, can you copy that into our SOP for pacemakers? Yeah.
Okay.
So there is a
yeah.
login kind of here. How about this might be easier. Let me download it and then I'll link the downloaded PDF.
Okay.
Because there's like a login wall that only a Feinberg student can get through.
Oh, okay. So Juhi, if you can show me your face as well through your phone. So yeah, 10 is a lot, but maybe Stas, talk to your friends. I told Eugene, Kim the same thing. People from our lab can talk to, people first about the movement and maybe Grace from our lab when she starts M1. She can talk to her friends, and maybe we can get 10 people interested and we can start something. I think we need to recruit first to register, but we'll see. We'll see what we can do. But
So the PDF is now in the chat for
Okay.
anyone who wants to take a look and see what the requirements are.
no, I'm pressing.
No, but you have to. Thank you.
Yeah.
Yep.
That's not the one.
So this is the right one.
No, that's not.
Okay, so I'll read. But Stats, on your end, just I think Easter has made a packet about like introducing the pacemakers movement and how to get involved, and there's a registration form, and a YouTube video, all that. So I'll tell Eugene O, can you tell Easter to send that to Stats, and send Stats, you can use that to distribute to your friends or people around you. So if we get 10 people, then I think we can start the official registration process.
Okay. Yeah, I'm just in my dedicated study period, so I'm not really seeing like any of my classmates. I won't until May, when I start rotations. So I could talk to them then.
Yeah, you can talk to them then. But if you have a student reserve, like any email list, just send out the email just when you can.
Okay.
All right. Good. Going back to Shauna, any other updates?
I guess like Easter Sam mentioned like Instagram.
Yeah.
We're getting Instagram exposure. I guess after we get everything organized and recruitment, enough like staff and admin to monitor certain platforms, we can gear towards like Instagram has like a new feature of having reels, so it's like videos. So we can use that to, I guess less work would be using in the YouTube videos and short clips and highlights of that to lead into like getting the viewers like hooked into going to our YouTube channel as well. So that's a feature that we can use. And then other video ideas for YouTube would be like maybe a day in the life. So someone would have a camera with them and just vlog their day in the life, and their like it's kind of like an in-person patient interaction, and those that consent to having that interaction too. That could be a little like less formal video idea.
No, I
I
like
think
that.
that's all I
Yeah,
have.
yeah. I think, yeah, I like that a lot. And then we talked about, Juhi mentioned humans in New York or of New York.
The pictures.
Yeah. So we're trying to take as many good photos during our interview. So there are a lot of photos, and then during the interview, we'll get quotes. So it's the picture and the quotes. We'll get a few series of them for each interview so that we can utilize them for Instagram.
Why is it two?
And then I think we got 3 interviews. I know Seungmi is working on editing, but Shauna, you can work with Seungmi to, edit the video and add the thumbnails and everything. Yeah.
Sounds good.
Have you seen them yet? Not yet, Shauna? Did
The
you?
videos, not the new ones, but I did see the first few videos that we were working on before.
Yeah. So we did 3. Yeah, let's work on them. And then Albert Curie, did anyone see the news? Albert Curie, double lung transplant, Northwestern. Yeah. So he's now famous. And we did interview him, even before this. So I think now, because people are going to, Google search, YouTube, double lung transplant, Northwestern, Albert Curie, or even my name, we're getting referrals from around the world. So I think it will be a good venue to also have people watch pacemaker videos. So I think his video is our priority. Let's upload it in YouTube in next week, in a week. Let's have, because it's such a timely shooting, right? So Eugene, work with Liam to organize the team and set deadline for each videos. Yeah.
Are you gonna split group?
No.
Aw.
Okay. Anything else, Shana?
I think that's it. I'll just talk to Sally so we can meet those deadlines and get people into like teams.
Okay. Great. Thank
Why is that-
you.
Thank
What?
you.
Papa, why is it
And
like-
then, okay. Dean, do you have updates?
I think, yeah, so since the submission process for the abstract, well, first, thank you for the comments and stuff, and we're able to submit that on time. I haven't been able to talk to Dr. Bumi since we submitted that. I did share a link to the manuscript with Dr. Eugen, but yeah, I know we walked through it a couple times last time, but I think at this point we're trying to decide which journals or which journal to submit to and how to adjust the manuscript to fit those formatting guidelines. So I know you mentioned JTOCRR last time.
Yeah.
I think just the only concern we had with that was the fact that their word limit for case reports is like 1,000 words, and our current manuscript with all the case report info plus the discussion is like around 2,000, I want to say. So I know Dr. Bumi mentioned BMC cancer, and I'm personally not super familiar with that, but it sounds like they have not as many restrictions on words. But yeah,
So
we're welcome to any suggestions.
BMC Cancer accepts case report for sure.
It sounded like from Dr. Bumi's standpoint that they were open to it, and I was just looking through the website, and they say that they broadly accept reports, and they did mention like new interventions type of stuff. But I can double check with her and follow up with her again.
Yeah,
Or-
index.
yeah.
Yeah, index. So the journal has to say we accept case reports. If they don't say it, it's questionable. So in that case, you email the editor and ask.
Yeah.
In any case, it's okay to submit to BMC Cancer
Okay.
and go from there.
Yeah, I'm just looking at the website now. They say that all presentations of case reports just must have consent for publication,
Wow.
but they do accept it. So
Hmm?
I think they might have their own consent form, but they also say they welcome whatever our own institution's consent form is for just getting that permission.
Hmm.
But I can follow up with the editor just to double check that. Yeah, that
Okay.
helps. But yeah, I think my plan is to then reformat the current manuscript we have to whatever sort of guidelines they have, making sure like the word counts for the titles and stuff and figures lines up.
Mhm.
And then I can send that new link to Dr. Eugene, but I can also come into lab and get other people to review that if that works too.
Yeah, the final, you should send it to the authors before you submit. So everybody, if you're submitting on behalf of all co-authors, you have to send the
Dad,
manuscript.
The pottery barn is here.
Okay, go and check. Maybe someone else.
Oh, No, I'm not gonna check.
So yeah, and then give them at least a week. And usually it's, I mean, if you have a deadline, it could be a couple of days, but you have to do that. Otherwise, you're not meeting the requirements.
Yeah, of course.
Okay. And then remember there are 2 clinical trials that you're helping.
Yeah, I think so. At least for Dr. Elena's project, I remember the last email communication was, I think, asking you to fill out a certain form.
About which project is this?
This is the Nivo viliparib trial.
Uh-huh.
And so the last email that I have in the email chain,
Uh-huh.
there was a form attached asking to submit a data release review before they can pull additional data for the statistical analysis. I wasn't sure if you saw that, but I can resend that to you if that's helpful.
Okay. Yeah, forward that email to me because I don't remember that email.
Okay.
Papa, don't use pen so hard.
Okay. So that's
and then but I'll make sure to follow up with
that's
them again.
that's with Dr.
Elena.
Stop
Yelena.
it, Daddy.
Okay, so let's move that forward. And then what
Yeah.
about Pedro? Is a Nivo Foreman.
Yeah, Pedro, we still haven't heard from, but I can email him again. And I think I have his phone number somewhere, so I can try calling him just to try
Yeah.
to speed things up. Yeah.
Yeah, let's move that too. I think we're almost there. So yeah, let's get that out. I think we need to sit down and see what has to be done. But anyway, Pedro and Jan has to tell us what's lacking.
Yeah.
Yeah, but it's been several years. It's so hard to move things. But yeah, you have to be persistent.
Yeah.
Email them every week to see
Papa,
where we are, and always CC me.
Papa,
Yeah.
who is this person?
What else? Is that all, Dean?
Yeah, I think that's all the updates, but I'll plan to touch base with Dr. Bumi so that at least this manuscript case report we can try to submit pretty soon.
Okay.
And yeah, and then I'll just stay on the lookout for other projects and stuff.
Okay. Good. Eileen, do you have any updates?
I think my biggest update is the Mr. Albert Curie video.
Uh-huh.
I made like the rough cut edit and then just put like the intro videos that like Shauna made and like the outro parts to it to the front and the beginning. I think I just have to work on the editing now, like the official editing. And just to clarify, you would want like subtitles, right? In English and in Korean. Do you think?
Yes.
Okay, yeah, and then I can work on that. I might need some help with the Korean though.
Yeah. Maybe
because-
ask Grace. Grace
Okay, yeah,
would be a
I'll do
good
that.
resource.
And then, yeah, I think that's my biggest update.
And so for his video, did we decide to split pacemaker and grapevine ministry, or where are we?
I don't think we completely decided that, but for what I did so far, it's just all together.
All together. Okay.
And then there were like some of his answers were a little bit repetitive, and so I like just took out the question itself because I didn't think it was necessary. But I do have the clips if we want to put them back in. But I thought, and also a lot of his answers were already like somewhat faith-based in a way. So I don't know if like it's possible to split.
Okay.
Yeah.
I'll leave it up to you. If you think it's not possible, we'll just leave it.
Okay. But yeah, just like even like the non-faith-based faith-based questions, he answered with something related to it sometimes. So I don't know if we could separate.
Okay. Okay.
Is co-host
So
better than co-host?
they're saying.
Then why is there a host and a pro?
Co-host. Yeah, so let us know when you have the final version, and then
Mhm.
well, actually before the subtitle, maybe you can show us the clips before to
Mhm.
send it to Liam and Eugene O, and then
Yeah, I did send it to Dr. Liam,
Mhm,
but
okay.
yeah,
Send
could-
it to
I need to
Eugene
edit
O.
it more anyway, so I'll send him an updated version once I do that.
Yeah. So I can give you feedback.
Yeah, I will make a Google like Drive that we can share the raw file, the image file, the video file, so that, and then we can upload as the edited version as well. Yeah,
Yeah.
I'll,
Also, there is one already through the Pacemakers Google Drive that we made a while back,
oh,
which has
okay.
like Zoe
Yeah, we can
and
use that.
Will's videos. So I can send you the link to that.
Yeah.
as well.
Please share
yeah.
with that one.
Yeah.
with-
So yeah, tomorrow I'll watch it with our lab members, and I'll give you comments. Yeah, just and then you can add the subtitle and do all the other editing, but
Yeah,
Who
now
is
it's just a very rough cut.
your rough cut. Yeah, it's okay, but well, I want to look at the final cut. Like I'll leave it up to you what to take out. But how long is it?
I can check. I think it was a little under 10 minutes though.
Oh, okay, that's
Yeah.
fine. Okay, yeah, let me look at that, and then you can start the subtitle and adding thumbnails and yeah.
Give me your hands.
Okay. All right, thank you, Eileen. And then are you working on any other projects or just pacemakers?
I was working on Pacemakers and then Grace's project that she was leading. but
Yes.
That's it.
Yeah. So work with Grace now with the new semester.
Yeah.
The goal is to write that into a manuscript together with
Mhm.
Grace. Okay.
Mhm.
And for that,
Wait, what?
you need to do more than 10 variants
Yeah.
for each gene. You need the
Right.
full list. So you need to work on that together. Yeah,
Who is he?
Good. Thank you.
thank you.
Papa, who is he?
I don't know. That's probably Eugene O.
No, Eugene was right here. Then who's this?
Lena-
Oh, I'm sorry. I'm using two accounts of
Two
your
of-
father, so that may confuse you. Sorry.
Using two computers. Okay.
Oh,
If you use my account to log
then
in.
your name is, what is your name from Emily? Dot Che.
Okay. Stas, updates from you. I know. When's your exam?
April 19th.
Oh, so it's pretty soon.
Yeah, and
Is
no
it
less than a
step,
month.
step one?
It's step one.
Yeah, step
Yeah.
one is the most challenging, I think. It's just a lot of, committing to memory and
Yeah, it's just a lot of stuff I have to like relearn from last year.
yeah,
'Cause it's been, a year.
right.
But yeah, it's not so bad. It's just a lot of and reviewing them. And yeah. So let me share my screen. I want to talk about a few things. The first is that maybe like a month ago, month and a half ago, I got an email about this workshop
Uh-huh.
right here. And they asked me if I'd be interested in applying, and they found my abstract via CITC. And so I applied, I got accepted, and I guess it's basically a conference, it's just smaller.
Mm.
And I'm gonna present it virtually, and it's the same poster.
Oh, so it's the same poster that they want you to present in a
Right.
smaller meeting too?
Yeah.
Both?
No, it's just like, I'm guessing that, I mean, this is a conference, but it's a smaller conference than CITC. It's
Uh-huh.
called a workshop. I don't know what workshop really means in this context,
Hmm.
but they wanted me to apply. And so I thought it would be another opportunity to present and meet people. So
Oh, yeah.
I did it. I've
Are you
accepted
gonna
it.
go?
No, it's like right after my exam, which is not the most convenient time, but yeah, it's really tight because I have my exam the 19th and then school starts back up the 24th and I'm taking my exam
Oh.
in California. Anyway, the point is that I'll at least be there virtually.
Yeah, yeah. No, this is great opportunity. And SITSI has a lot of, yeah, this is great. The more presentation you do, the better. And there's a lot of award opportunities too. And then they
Great.
invited you to present. So that's, you could say you
That
were
means something.
invited to, present, and that just is an honor. And people are interested in your topic.
Right.
Yeah, yeah. So great.
Okay, so that's the first thing. The next thing is I wanted to provide some updates on where the
Papa,
squamous cell adeno TMB ITH project is. So here, so I'm going to show a few figures and I just kind of want some advice on the thought process to take. Just to kind of give you some context, I had to make some changes to the way we were running PyClone as I learned how to run it better. And so when I did, I generated kind of a new data set. The new data set still has the same trends, but the cutoffs are different. And so really what I want to focus on is selecting these cutoffs in a way that's most appropriate. So what you see here is a plot of kind of three variables, the two cutoffs, TMB and cluster number, and p-value. And this is for squamous cell. And the color of the dot is proportional to the sample size. So the darker the dot, the higher the sample size. So I looked at this for squamous cell. This is what I got.
Hey, Stas,
Yeah.
sorry, I have to answer the door.
Yeah, no problem.
Let's do a 10-15 minute break because
Sure.
it's been yeah, sorry about that.
No,
I'm
that's fine. No
working from
problem.
home today.
Yeah, no problem.
Okay.
Okay, so what I'm showing you here is a plot of the cutoffs with their associated p-values. The points that are on here are of a p-value less than 0.05, and the color or the intensity of the color of each dot is proportional to the sample size. And so I wanted to first look at squamous cell, and then I also looked at adeno. And so I guess with squamous cell, if you just briefly look at it, you can see that there's clearly a cluster down here that has pretty high sample size and has a p-value of a less than 0.005. On adeno, this group right here has the lowest p-value, but not necessarily the highest sample size. But I think this kind of all comes together a little bit when I plotted both of these together. And so these are the two overlapping groups. So originally we had selected a cutoff that gave statistically significant relationships in both adeno and squamous cell. And originally I thought that was kind of the correct way to go about it. But if you just look back, at least at squamous cell, you find that the p-values are significantly lower for cutoffs that don't overlap between the two groups. So I plotted this all together on one plot. The blues are the overlapping clusters or overlapping cutoffs. The red is unique to adeno, and the green is unique to squamous cell. And so in the discussion in the cutoff selection discussion, we have to ask ourselves, I think, are adeno and squamous cell heterogeneity distributions the same? Can these molecular biomarkers kind of be treated as equivalent? And that's one of the points I kind of wanted to make in this paper, because it really goes along with all of the other evidence we've seen. All the other molecular biomarkers have shown that these two groups are very different, yet in clinical practice, the cutoffs that are being used, at least for TMB, are exactly the same. And so you can make the argument that by selecting cutoffs that are specific to both groups, you get higher p-value relationships, more statistically significant relationships, in some cases at much higher sample sizes. I then went a little further to see if the microenvironment was different between these two types of cutoffs, the overlapping cutoff, which is the blue, or the unique cutoffs. These are the cutoffs that I chose just to kind of test this idea out. The overlapping cutoff is 9.4 and 3.2. And then these are the two unique ones, which give you significantly lower p-values. So when I look at the overlapping cutoffs for squamous cell, the Kaplan-Meier survival plot is on the left. And on the right are the microenvironment cell types that are statistically different between the high heterogeneity group and the low heterogeneity group. What's interesting is when you pick the unique cutoff, there is significantly more, or not significantly, but there's very obviously more cell types that are different between the two groups. Yeah, and so I did the same thing for adeno, where the same was true, but not as big of a difference. But honestly, the point of this is that I'm really struggling, or I have been struggling, to really find like a unifying thesis to the paper. And I'm thinking that maybe the right way to go about this is to say, hey, look, we found this relationship. It shows that from a molecular perspective, adeno and squamous cell are very different. They have different relationships. They have different prognostic value. And this goes along with all this body of research that has shown that molecularly they're different. Yet in clinical practice, we treat them as the same. Kind of the end. I don't know.
Right. So go back to the So
this is adeno and this is squamous cell.
Yeah. So starting with squamous, low TMB per our own cutoff.
Right.
High ITH portends better prognosis.
Correct.
And then the next adeno. And
that's adeno.
Yeah, and
with
adeno,
the same cutoff.
Yeah, both 9.4. Okay, same cutoff. Cyclone, same cutoff 3.2. Both same cutoff. High ITH actually portends poor survival. And have you looked at overall survival too, or just PFS?
I looked at both. OS is also significantly different. I just don't remember the numbers. I can pull it up.
Yeah, but for both, or they're significant for both?
Yeah.
Okay.
I'm pretty sure. I'll double check, but I'm pretty sure they were.
Yeah. So let me understand what you did. Go back to the very first graph.
So I wanted to basically plot all of the cutoffs that we're interested in, kind of the ones we can select from. And I thought the important pieces of information were sample size, which is indicated by the color, and the p-value.
So sample size meaning, so let's say if you have, I want to understand. So if you do TMB
sample, it's the sum of both of both sample sizes of the high ITH and low ITH.
So I can understand, because our graph is only looking at low TMB. So if the cutoff becomes higher, then it's going to be, darker for sure.
Right.
For a cluster number, I guess it doesn't change much, right? Because we're going to have high ITH, low ITH. You know what I mean?
Right. It seems like there are more samples with a lower cluster number. Although there are samples here that have a cluster number of say 3 or close to 3 that
Mhm.
are pretty dark, so around 300.
Yeah. Yeah. So I guess cluster numbers don't really matter
No.
when it comes to the sample size. Now the p-value is determined by the Kaplan-Meier p.
Correct. Well, it's called the log rank test. So
Log rank.
it'll look at both groups and say, what's the odds that these two distributions are statistically different or the same, really? What are the odds that they're the same?
Right. And in your definition, what is the ideal cutoff? Like looking at the graph.
Well, ideal. So if we look at kind of the overlapping cutoffs, these blue
Right.
ones,
Uh-huh.
and then I think the ideal cutoff is a unique cutoff for both groups,
Mm.
which goes, I think, along the line of the argument that they're molecularly different. So we
Right.
should be using different cutoffs.
So looking at this, like what is there an equation to drive cutoff or
Well, I mean, what I did is I took every single cutoff and I looked at what the p-values were that the cutoffs would give.
uh-huh.
And so we just have to figure out whether it's p-value, sample size, differences in expression, or differences in cell type prevalence. What is our primary or preferred biomarker? Like, or not biomarker, but what is our evaluating criteria? What matters to us the most?
So how did you drive 9.3 or that number?
How did I choose these?
9.4, yeah.
Oh, I looked at the graph to see where I had the highest sample size and had the lowest p-value for both subtypes. And so what ended up happening is the p-value is really similar at 0.023 and 0.024 for these two cutoffs. And I picked a higher 9, I picked 9 as opposed to picking one of these lower values. I picked something closer to 10 because 10 is the cutoff that's used clinically.
Right.
And so what's interesting is with squamous cell, 10 and 1 is a really good unique cutoff, which matches kind of 10 as the cutoff that's used clinically. But in adeno, a better cutoff is 4.8 and 3, which kind of puts it clearly is clustering very far away from how squamous cell is performing.
Mm-hmm.
And what you're seeing here is everything that's lower than a p-value of 0.01. Because it's performing quote unquote better than the overlapping cutoffs, because here I kind of prioritized p-value over other things.
Okay. So I think, well, I like this approach. I think if you can explain it better how these numbers are derived.
Right. So the cutoffs, I mean, those we just get from TMB we calculate, and PyClone generates the cluster number.
Right.
Now the p-value is just, we create two groups. What are the odds that these two groups are the same? And then the sample size is what's the sum of both sizes, the high ITH group and the low ITH group.
Right. And how did you define overlapping?
They were the same. So the cutoffs were the same in both subtypes, and both cutoffs yielded a statistically significant result, then I would consider them overlapping. So really what it's saying is, can you pick one pair that works as a cutoff for both subtypes?
And working means less than 0.05.
Correct.
Okay. Okay. Have you seen other groups work creating similar graph, or is something you thought of?
Well, when we were doing when I was writing the heterogeneity review, there was one metric called allele. What was it called? Wow, I'm blanking on it. Allele heterogeneity frequency, allele frequency heterogeneity. I don't remember, but the point is they plotted their cutoffs with hazard ratios and then used that relationship to pick the optimum cutoff.
Hmm.
So hazard ratios really kind of capture a similar metric, more outcome related than you know, are these groups different? But
Here you got p-value. Right?
basically, yeah.
Okay.
So that's what I've seen. But most papers will say, oh, the FDA says 10 for TMB, that's what we're going to use. But that might not be the case when you actually split this up into subtypes.
Yeah. No, I think this is unique because I haven't seen this in other papers, but I think it scientifically makes sense. But to get to 9.4, would you say, I mean, would you say lowest p-value? So overlapping cutoff pair 9.4. Would you say that the 9.4 and 3.2 and that p-value is coming from the lowest p for overlapping, or what is the driving?
Let's look here. So the lowest one, the lowest p-value is probably around here. And that's like 0.01 to 0.05.
But then it's not 9.4.
It's not, right? So these are these. I could also pick those and try and work with them. I only picked 9.4 because it was closer to 10, which is considered kind of like the gold standard because of the FDA. I could
I
pick
see.
these and look to see what they look, how it looks as well.
I see.
It's just a matter of deciding what I think makes the best argument. Because I think if we argue like, hey, we wanted to pick an overlapping one, so we picked what was closest to what the FDA said, and this is what it looked like. Now we tried our method, which focuses on unique cutoffs for each subtype. I don't know. I mean, I'm very much open to suggestions.
I see what you mean. Okay.
Like this finding we have is interesting. It's how do we tell the story in a way that is believable and lines up with all this other evidence we've seen. And I think it does. It's just, yeah, how do you tell the story? So that's why I was thinking of saying, hey, we have these cutoffs, they overlap. This is what we get when we use an overlapping cutoff in terms of the microenvironment. And then when we pick specific unique cutoffs, we get something different or something more powerful statistically.
Yeah. Yeah, I think so in a way, when we say high TMB, low TMB, it's determined by, let's say, lung squamous, lung adeno. And what is the actual median of TMB? Are they similar or same? Similar or different? So that's easy to calculate, right? And what's the median? What is the distribution difference of TMB? And
I mean, yeah, that's part of it, right? Because then you have to ask yourself, well, let's say the distributions are the same, but the relationship between these values and outcome might still be different and wouldn't be captured by median or the distribution of the value itself.
Yeah. So I think the point of your manuscript is to show how squamous and adeno are different in lung cancer. I think showing the distribution difference between TMB itself and then the cluster number itself, just putting those maybe in one graph, you can have one plot distribution for squamous, one distribution for adeno, for y-axis being TMB, and another graph,
Yeah.
same two graph with one axis being cluster number. So I think you should produce those two graph at the least.
Yeah.
And I still think this graph is meaningful, so I think it's okay to add this. And then, but I think the convincing the editors that, you know, with these three categories, how did I pick the one that I want to represent?
Right.
And you could go by the lowest P no matter what, or you could go by highest sample number. But if you go with high sample number, you have to go by the highest TMB.
Right.
So I feel like we have to have a certain logic, not just pick one that's close to 10.
Yeah.
Then there's no point of
Right.
drawing this graph. So you could argue that maybe go for lowest P for all three groups. And then also show, you know, so that's three Kaplan-Meier curves, both PFS and OS. So that makes it 6. But you could do, I mean, regardless, you just choose one that's clinically meaningful, which is 10, and you could pick any, you just say you chose anything that's closest to 10 as a
Yeah.
cutoff, and then choose the cluster number accordingly on 3 groups. So in that way, you can kind of justify, you know, like
Right.
you pick the p-value as the, you know, defining rule, and then the fourth is clinically, I just chose any value closest 10 for each group, and then show such graph. So I think the whole point is, you know, by cutoff, they're different, and it's the squamous and adeno are very different. No matter what kind of cutoff you choose, they're showing the opposite
Right.
finding. No matter what kind of cutoff you choose. I think that's the message here. So yeah, why don't you make those graphs for the actual manuscript?
Okay.
Okay, let's do that.
And then kind of the secondary graphs will also be, not graphs, the secondary tables will just include all of the scores that I've calculated, the FFA, the estimate scores, and then how do I kind of, what's the right approach to making sense of these TME differences, these microenvironment differences?
So I think this is good. I think you should just maybe choose one cutoff to show the stark difference.
Right, but then if you choose a cutoff that's kind of overlapping, that works for both subtypes versus the ones that are unique, you get different relationships between well, not necessarily different relationships, but you get more statistically significant differences in cell types.
Yeah. So yeah, I think you can do it just for unique cutoff for this one.
Okay.
The whole point is, I mean, immune distribution is very different between squamous and adeno.
Right.
That's all. You don't have to say, you know, you don't have to try to explain why. You can't.
Right. I guess I meant more like
You can just.
Yeah. Go ahead.
Yeah, you can just. I mean, you don't have to explain, but if you find any literature that backs up our finding, yes, some other literature did find that in squamous that these cells were more prevalent or something, just back it up. That's all you need to do. And we don't have to say
Okay.
why.
Right.
Yeah.
I guess the point I wanted to make with this is if you choose a more if you choose a unique cutoff with a higher p-value, you not only get a higher p-value for outcome, but you also get a higher p-value when you compare the cell types. So I guess the argument, more specifically, the argument is unique cell types allow you to generate more unique TME groups. But
Yeah. No, yes, I like that message. So
anyway, that's it for this.
okay.
Next, I wanted to show an update about Tempest screening. So I have this sheet that I've made, and I've begun to screen as many of these as I can. We have 96 with Gardent, that about half, or no, a little less than half, 43 have been screened. And when I say screened, I mean diagnosis date, treatment start, end dates, treatments, and then this is empty, and then the last encounter or death date. And then this information we can use to then try and mimic the same sort of analysis, look at PyClone, look at TMB, look at outcome, and then we can also add more samples to EGFR, our EGFR abstract, not abstract, but the hypothesis about EGFR TKIs.
So
yeah.
explain. So this is, I see Tempest, but are they also Gardent? Like, is it
So-
overlapping or?
So in the first 96 or 95
Uh-huh.
have Tempest and have Gardent. That doesn't necessarily mean that the Gardent that they have has a matching date as the Tempest, or was before or was after. It just means that that patient has at least one Gardent sample throughout their treatment.
Mhm.
And then we have another almost 200 of accession numbers and IDs from Tempest that don't have an associated Gardent, or these patients don't have an associated Gardent test. And this is another set that should be screened. Unfortunately for this set, all we have is the birthday and the initials.
That's just the Tempest?
This is just Tempest, yeah.
Okay.
So my plan was to just start with these first 96, get through them,
Hmm.
and then decide on how to proceed with the rest.
Yeah, I'm sure if so is that the Tempest accession number? The one
Correct.
next to it?
Yeah.
Yeah, I'm sure they can give us the names.
I asked them, so originally they just gave me the accession number and the initials. And then I asked them for more information, and all they could provide me with was the birthday.
Birthday. Okay. Is there a way to find them? If
Yeah, I think if we type in initials into Epic and the birthday, it'll give us a list of patients. And then we have to pick one out of that. Hopefully that list is really short. We also know the physician associated with those patients, so we can check to see. So it'll take a little bit more work, but it's doable.
Okay. Okay.
Alternatively, something that I haven't explored is maybe if you type this accession number in manually into Tempest, it'll tell you the MRN for this patient.
Oh,
Maybe, I don't know.
okay.
But that would be manually, and we can do that. Like, whoever, when you screen this first, you go to Tempest, you type this in, you get the MRN, and then you type the MRN into Epic.
Yeah. No, so
ahead.
getting access to, getting those Tempest access and building these patient outcomes. I want you to pair up with Arnie here. She's right here. Because she's gonna be in our lab for 6 months, and she'll be able to work with you to pull up all these data.
Yeah, that would be really helpful.
Yeah, and help you as much as she can. So why don't you why don't you have a separate Zoom meeting with her, and Eugene, oh, you can attend as well, and kind of go through, how to access the Tempest homepage and get those data, and how to look up these patients in the Epic.
Yeah.
Yeah, and then she could, right now you have to study, but in the meantime, she could work on getting these patient outcome data. Yeah, let's do that.
I definitely have time to do, a few a day. It takes,
Uh-huh.
in my experience, it'll take me anywhere from 10 to 20 minutes to do one, just
Uh-huh.
because you have to really read through the chart and
Uh-huh.
figure out what the treatment was, why were they were taken off.
Right.
So there is a time investment, but
No,
I
let
can-
Arnie be involved and work with you.
yeah, absolutely.
So,
Yeah, as many people as can help is, the better.
yeah.
Because, even in my kind of screening, there's just a lot of patients that fall off. So for example, like even though we have data for them, some patients, their chart will end in 2017
Mm.
and they probably sought care elsewhere, but we don't know what happened to them. But we
see.
know that they at least survived till 2017.
I see.
So
Yeah, Arnie is a physician working. So yeah, she'll have more like clinical knowledge to
great. Yeah, that's
that
perfect.
data. So let's talk about the whole purpose of this project.
Right.
I guess the whole big picture is, for those of you who are new to this project, we're trying to analyze tumor. I mean, we know what mutation burden is. We know tumor heterogeneity is, but it's so hard to assess them before they die. After they die, we can autopsy and did what Charlie Swanton did, like autopsy and biopsy every single metastatic site.
Right.
And then you'll have the map, genealogy tree of the heterogeneity of all the tumor metastatic sites. We can't do that when they're live. And when they come to see you in clinic, we can only rely on two things. One, tissue biopsy, one, liquid biopsy. And from that, what's the best way to understand their tumor heterogeneity? And how would that be clinically relevant in terms of guiding our treatment? That's our big question. And what we have is Guardant data. What Guardant, they have graciously provided us the clonal numbers, and then they have blood TMB numbers. Tempest gave us raw data. So Stas, you were able to get the, well, first of all, the TMB, did you use the number they calculated?
Yeah, so this TMB number, so where it says the number and where it says NAN, so if you see PyClone, they have a TMB value. It's just, it requires us to manually go in and check it with the report, which I can do, but this is their number. This is the number that they calculated from the same tissue as this raw data came from. So yeah, it's their TMB.
So Tempest State PC is PyClone?
Yeah.
So how did you calculate PyClone in the Tempest, from the Tempest?
I took their raw sequencing data. Well, it's not raw, it's processed, but the point is I took the read counts for the different alleles from one set, and then copy number, combined those, and then ran it through PyClone, just like I did with TCGA.
Okay, so same way. And
yeah,
was there a difference between, because this is like 600 gene panel, TCGA is whole exome. Was there any difference?
In what sense? I haven't compared the two.
Okay.
I just haven't. I guess I could compare the distributions maybe,
hmm.
but
Yeah, whether running it in XSOM versus a this, what do you call this, the selected panel or hotspot panel, or I don't know, I forgot. But anyway, that this is best we can do. And to get the PyClone number, don't you need like cutoff it there as well?
you need enough mutations. So
Mm.
I don't know what the minimum is. I think it might be like 30 or 15. I don't remember. It's not a parameter that I have ever played with within PyClone. It's
Okay.
just it's a built-in default parameter. But
Okay.
as far as I remember, most of these, if not all of these samples, had enough mutations.
Okay. Okay, good. It seems like it's either 1/3, 2/3, or
Yeah, the reason
prime
it's the
number.
thirds. Yeah, the reason it's the thirds. So the way that I did this, so when you run PyClone, it
Mhm.
kind of starts randomly as it clusters. So day to day or run to run, there will be slight differences in the clusters. There might be 3 clusters one day or 2 clusters another day of the cluster that it's selecting is a little too small. That third is too small. And so for TCGA, I ran it 9 times, 3 times on 3 separate days to cover kind of the randomness. Here, I only ran it 3 times. I didn't do it 9 times. And so that's why you're seeing the thirds and 2/3. Yeah. But when I run it 9 times, that will average away. I didn't run it 9 times because 9 times was just, it computationally takes a while. And this was just, I was just trying to prove some concepts and that I could run the pipeline.
Okay. Okay. I know.
Because the reason I do that is because I learned that if you don't do that and you don't average out, then you'll run PyClone and then you'll get these values and you'll have an amazing relationship. Someone else runs it and they'll never see that because it's, but I'm thinking that if you do it enough times, you can approach some kind of consensus.
Yes. And do you find it sometimes very different, or are they kind of close to target?
They're close, but if enough samples within the sample set are different in like, you know, if enough are different in the low cluster number direction, then it can sway the, it's literally random. Like it really just depends on how close to the consensus each individual one is. If you get enough that are really far from the consensus, you'll get a very different result. I mean, the trend will be the same. The p-value will be different. So in some cases, you can get a p-value that's 0.002 between the 2 groups and then 0.002.
Nice.
What I'm trying to say is I'm hoping that running this multiple times averages out kind of the randomness and approaches whatever true consensus exists.
Got it. So have you run anything from the initial Tempest clone and TMB?
I haven't, just because it's a fairly small sample size. So right now I have 43 that are annotated. Of
Okay.
this, I just need to manually go in and fill these in from the Tempest document.
Okay.
But even when I fill them in, 43 or 42 is pretty small. Also, I think it makes sense for us to maybe group them by initial treatments or I don't know how else to kind of normalize them or normalize the differences. Also, a lot of these aren't all squamous and adeno. There's some poorly differentiated ones, and I think I saw there's a large cell.
Oh, I see. Well, yeah, it doesn't
And small
have to be
cells.
either squamous. I don't know. I think sometimes we want to just group them together for different projects, right? I mean,
Right.
we still need to collect everything. Yeah, so even just simple things as getting TMB values, just work with Arnie because she's
Right.
in the lab. and she could definitely help you with all these.
I think given her background as a physician, it makes more sense for her to screen the patients and
Uh-huh.
then manually entering TMBs. Like I can do that. I don't, or
Yeah, that's easy, right? You
yeah.
yeah. But yeah. Okay. So those are so I guess the simple task would be to do the landscape of all each variables, and then in Tempest sort of like in real world data, right? And
Yeah.
the the, the in I guess what I'm trying to get is uh what the relationship you've just shown with your graph was in early stage. That's all TCGA data, but this is
Right.
mostly stage 4.
Yep.
And the question is, we believe the molecular landscape is going to change as they advance to a higher stage.
Yeah.
So the Tempest data is great to ask that question. Would that kind of a relationship still hold with advanced stage?
Right.
And we
So
can
I
ask the
think,
question, right?
yeah, no, so I was gonna say that once I have the early stage manuscript written out, I can try and see if I can extract some sort of relationships from the late stage. I just remember the associations weren't as strong, but maybe if I approach it in the same way that I'm approaching these cutoffs, maybe I'll
Right.
find something that we can
Right.
I don't know, write an abstract on or
Yeah,
something.
we don't have much sample size yet, but I believe, ever since we got this data, I think I've screened more patients with Tempest, so I think we can get more data.
Yeah.
And why don't we start that now? I mean, I think I did CC you on my previous communication with Tempest.
Right, right. So I've been talking to them and there is a lot of data. So, other than the overall, other than the patients that have both Garden and Tempest,
Yeah.
there's a total, basically there's 309 patients with Tempest. Of those, 95 have Garden. So there's 200
Okay.
that at least have been screened in 2020.
And are they all lung or are they everybody?
I think they're all lung.
Okay. So we did ask
But they're
for-
not only your patients, they're also patients of other physicians. So I don't know what that means for the IRB, but
No, my IRB covers every lung cancer from the
okay.
institution, so it's fine. Yeah, 300 is not a bad number to look at landscape.
I mean, we'll definitely have to filter them because of those 300, they're not all adeno, they're not all squamous. And then some disappear.
Right. But
like-
as far
these
as
guys.
collecting data goes, I think
Yeah.
we should collect everybody just
Yeah,
because
yeah.
of the heterogeneity project.
Absolutely.
But then once we collect everyone, I think you can ask that question. Yeah, in advanced case, do we see that similar difference or not?
And then there's also one other question that we talked about answering with this data set, and that's looking at the treatment time
Yeah.
for specific TKIs. So that's kind of an accessory question that we can try and answer as we add more numbers.
Yes, absolutely. Oh yeah, so Stas, you did e-poster for AACR?
Yeah, it's just, it would have been really difficult to travel over there while studying for STEP and expensive. And so I just did the e-poster option.
And was that the EGFR?
No, this was, so for AACR, I did lung adeno.
Oh,
I described this lung adeno relationship with TMB and PyClone. And
okay.
then for ASCO, we submitted the EGFR TKI, but our sample size was small.
That
But
was
I haven't
ASCO.
seen,
Okay.
yeah, right? AACR is the one that's in April. And then
Yeah.
ASCO is, yeah. Sorry, sometimes I get them
Yeah.
confused.
ASCO, we didn't hear yet, right? What's accepted?
I
What's not?
haven't. I think Liesel is the one who submitted, so I think she will probably get the notification, but I'm not sure.
Okay. Okay. And World Long, you didn't submit anything, right?
No,
World Long? Okay.
No, I was so busy with school
Yeah.
that I just,
So-
yeah.
That's okay. So I think the immune landscape and TMP, what if you ever do, ever explore this data set, that would be good for the SITC. That's coming up in November.
Yeah, that's a good time frame. I think by then we could definitely screen this and actually have something,
Yeah.
and
And-
to talk about.
SITC is in Boston, so if you ever want to go, I don't know what the time.
Yeah, I mean, the reason I went to SITC in Washington was because the school offered to reimburse me, but they
Mhm.
only do that. It's kind of frustrating. They only
Yeah,
do that for one conference all 4 years. So
Yeah.
it's like, wait, don't you guys want your students to travel? To
Yeah.
the conferences. But
Well, so I'll tell you, the World Long and SITC are generous in giving out travel awards.
yeah, I'll definitely apply. I mean, it
Yeah,
doesn't hurt.
yeah. So keep applying. I know ESOL got the travel award. And I know if you get the award, they pay for the flight and hotel, taxi, everything. So they'll
That's
cover
great.
you for everything.
Yeah.
And the registration, I think, maybe. Yeah. So keep applying. I mean,
Do you know if that's a matter of, is your abstract interesting enough, or is it, what is kind of the main factor that affects
I
the
think it's
decision?
both. Yeah, I think it's your sometimes, but well, I think it's quality of your work. But then as a student, if a faculty did this, maybe not of a big,
Right,
yeah, but at the student level, you did this as a presenting author, then maybe you get more attention.
right. Because Liesel was applied got hers as a resident, right?
Yeah, yeah.
So I was thinking, maybe they prioritize residents or more senior student. I don't know.
Yeah. So every conference is different.
Right.
And I think for the World Long, even Grace, she's undergrad.
Yeah.
And she applied, she was able to apply. They don't discriminate
Right.
undergrad, medical student, or resident. They all consider them people as a student,
Yeah.
category. So yeah. Anyway, I think this is great data set and a lot to explore, real world data. And now you can do the PyClone with Tempest, and then we'll just go into our original question of how do we compare heterogeneity from blood and tissue, and how do we integrate that into a one metrics.
Right.
So I think let's gather more data and really delve into that question. And I think a lot of other groups are looking into it. I mean, we've been talking about this for 4 years now. So
Yeah.
yeah, it's not easy, but I think we have lab members now who can work with you to
Right.
get more data and really brainstorm together. So Arnie will work with you closely. So just set up a meeting this week or next week whenever you have time,
Yeah.
Stas. And you can help and orchestrate all this. Okay, very
Great.
good. Anything else?
Not for me. That's
Okay.
it.
Yeah, we'll be happy to help.
Yeah,
Okay.
thank you. I really appreciate it. The more help, the better, because screening these things takes time, but it's very high value. And once we have this data set, I think we can answer a lot of questions. Ask a lot of questions.
Oh
and
yeah,
help
so
out.
Stas, send her your recent publications and the review paper, right? So that she gets into the this field of heterogeneity, and she
Right.
can start reading and get herself motivated. Yeah, so you can send it to her, the messenger, right? Everyone's there to find her and send it to her, and maybe to Eugino as well, because she'll be attending the Zoom call.
Yeah, absolutely. I think it makes sense for me to email that out just because it's
Oh, yeah. So
versus
Arnie,
sending it in
put,
chat,
put
but
your email in the chat box so Stas has it, okay?
and then I can send out an email later today with all of that.
Yeah,
Yes.
okay. Cool. Okay, so we'll move on to Seungmi.
Yeah, so let me share my screen first. Okay, share screen. This one. Yep, so with my project, we've sent an abstract with the help of Eugene and Juhi. And now we have to write the transcript. And I discussed the roles, and the graph and conclusion would be me, but I'll also be in charge of overseeing the project and make sure it gets done by this deadline we just made for ourselves. And Juhi said she's happy with doing method and results and introduction, and she has already started on it. She's gonna get it done in a few days. And Arnie and Shah, they are given the big role of doing the literature review that goes into the work of writing a good discussion. And Arnie, I've told this to Arnie, she's happy with it. I'll have to talk with Shah today. But of course, if anybody who is finding this too much of a challenge, Julie and I will jump in to make sure. So we're not just going to say, oh, you guys do it. So we're going to be also reading papers and help oversee the project. So that's
So
the
let me-
plan.
So I think Seungmi, you have to lead the discussion because it's your project, and I think Arnie and Sha can help you, but they don't understand the project like you do. They're not the initial member, so they don't understand the whole discussion that happened when we submitted the art abstract.
Okay.
So it almost feels irresponsible for them to lead the discussion. I think you have to take the lead in the discussion because to tell you the truth, above everything else, discussion is the core. Okay, discussion is what tells us this project means something, and this is where in the whole history of medicine, this is where our project lies. And this is the meaning compared to other projects. This is what's unique about our project. Okay, so I feel like the lead author has to lead the discussion. And because it requires a lot of referencing and looking up other things, you definitely can get help from other people.
Okay. Yeah. Okay. So I'll change that. And the graph is a big deal, it seems, with landscape, and we just want to get it done more professionally than the one we did last time, more refined. So I can't do that by myself, so I'll ask someone else to help me out with it.
So you can ask Arnie and Sha with graph and table, everything. And you can still have Arnie and help you with the graph, table, and discussion. But you have to lead them in terms of what they need to do. They can't create something new at this stage. You are the brain here. Remember all the time that you spend reading the literature, okay? For Arnie and Sha to get there, it's gonna take a lot of time, another 3 months for them to get to your level. So you cannot ask them to read all the literature and get to your level, you lead the discussion. Do you understand?
Yeah.
This is not just for your project. For any other project, when you divide and conquer, the brain part has to be the lead author. The lead author has to know all the literature out there, has to direct all the pieces. You can get help, but you cannot rely on other new members to write discussion. That is very irresponsible. I'm
Okay,
just telling you
totally.
the general rule, okay?
Yeah. Yeah.
And you can put
Um,
Arnie and Cha in graph and table.
okay. Yeah, and of course, Eugene's welcome to join anytime as much as she wants. And she's always welcome. Um, yeah, and so I did start
Okay,
studying.
one more thing. For any collaboration, the buck stops at the first author, okay? If anybody says you, oh, I'm so busy, I can't do it, you are responsible to write everything, okay?
Yeah, yeah.
If if they say, I cannot do it anymore, I have such and such things come up, and you are the one who decides this author has made some contribution, but not at the co-author level, then you put them in the acknowledged part.
Yeah.
And they will suffer consequences of not helping you if they choose not to.
Yeah.
Okay? So,
Yeah.
so I want you to understand that first author is truly responsible, and you cannot tell the PI, oh, well, this was supposed to be done by Army and Shah. They didn't do it, so therefore I cannot do this. No, it's your job. You're getting help. So it's not like
Okay.
you've delegated, but still you're still responsible. Do you understand?
Yeah, I understand yeah, I totally understand. I was kind of like trying to shift it to them and say, hey, maybe you could be first, I'll be first with it, but I understand that this is my project, and I will be, if like let's say nobody turns up, I'll be responsible for having to write it. Like in theory. So I
Right.
understand.
And then if it comes to a point, hey, I got something come up in my life. I gotta go to a funeral. I gotta take care of others. I cannot do the project, and you give it to me. And I take all the responsibility. I'll find other people to work on.
Yeah.
And you're not going to be the first author, and that's the end of the story. So it's almost like you carry it till the end. If you can't do it, you pass it on. The worst thing that can happen is you tell other, I will do it, and then every time I ask, I will do it, and then that happens for like a year. No one completes the project. That's the worst scenario in any project you do. So when you feel like anybody who's working on a project, when you feel like you're far stretched, you can't do it, you just say, I can't do it. And you say, I'm going to be available in next in 3 months later when I finish step 1. Okay. You have to say, otherwise the PI does not know. And then it's fine to wait till then there's no issue. But if it has to be finished by some certain deadline, then it's almost like a sin to hold it and not do anything about it, because you're just losing precious time. So it has to be passed on to other people if you can't do it. So I'm just not saying this to Singmi, I'm saying this to everyone who's doing a project. And I think it's very important that people know how to keep their deadline, what their role is. If someone else does not do it, whose responsibility is it? That's why becoming a manager is such a difficult task, because people quit all the time. Working in an organization where there's a high turnover, it's so hard. And Stats, you've seen our lab members changing every 3 months, and it's difficult. Like, and they leave, and then you got to work with someone new. but
Yeah,
now
I don't think anyone here was here when I joined back in 2020.
maybe Grace has been
Oh,
pretty
right.
Yeah. So the students get to stay the longest. I have researcher coming like for a year, a dedicated. She's a breast cancer surgeon. She's so much into genomics. She's coming solely to learn NGS. And she's a very prolific and very productive breast cancer surgeon. So yeah, so we'll have good members come in and be good for continuity. But still, we still welcome 3-month dedicated researchers. All I ask is your commitment, and when you feel like you can't do it, you tell us sooner and pass on your project. So so much for that. Okay. Go ahead.
So I did set up a deadline, which is I leave at mid-May. So if I have this deadline, then probably we'll get at least something done, and hopefully, I am trying to be serious about this. So other thing is nothing much to update other than Juhi's doing a lot of hard work with getting the method and results done, and I've also started literature research and started 2 courses in Coursera to help me understand bad data, and yep. So and the other
So
Mhm.
here's the thing. I could sit down with you guys, all of you guys, and if you come bring the literature and the key literatures, I can even verbally write a discussion with you. Like this is the first paragraph. You need to cite these, and this is the points. The second paragraph should be this. I can literally write the discussion with you at the spot. Okay. You don't have to like, oh my God, how do I write this? No, this is something that I can always help. Okay.
Yeah.
And it's just a day or two, you'll have a manuscript ready. So don't think of this as a 3-month project. If you have the abstract, if you have the results already, you should be able to write the paper in a week. It's just a matter of people helping, making the graph and table look nicer and putting the reference in the right format. If you get help, and undergrads can help you with those too. Once you have the abstract, once you have all the graphs in your mind, this is, to me, if you work full time, this is 1 week worth of work.
Mhm.
Okay.
Yeah.
So I want you to be curious about this and definitely submit before you leave Chicago. Okay.
Yeah. Understood. Yeah, so I'll set another deadline. So method and results, me and Julie will get it done within a few days. Yeah, because Julie's like a powerhouse when it comes to writing. So we'll get it done in a few days. And then yeah. Okay. And yeah, the other thing was I have presentations to do, but do you think I should do it now or wait until everybody got their projects discussed?
No, do your presentation.
Okay. Oh, where's my presentation? Um, oh, sorry, not this one. This one. Sorry. Yeah, so it will be it's like a 7-minute presentation. And it's very interesting. And it gives you some insight about the nature of research. So I'll start now. So this is like a short presentation on this journey, which eventually won the Nobel Prize in 2005.
So what is the
And
title of your talk?
sorry, what?
What is the title of your talk?
The title of my talk is so it's a short presentation on the process of discovering Helicobacter pylori as a pathogen and deeply involved in the etiology of gastritis, which eventually won the Nobel Prize in 2005, and the specific journey a doctor had to undertake to prove this to the world under a lot of oppression. So Dr. Barry
Wait,
J.
wait, wait, wait.
Marshall.
Before you present, I want to understand why you're presenting this.
Oh, the reason why I'm presenting this is because while I read about this, it was just more than finding out the cause and effect, the causative bug and the effect. It was a journey that could teach us a lot of lessons about starting from the science to establishing cause and effect and getting it known to established doctors and the world. And so that's the reason why I'm presenting it. It's also like you asked me to present it.
Did I ask you to present this paper? Did I ask you to present a paper?
No, just about this, like, because like this dog Barry J. Marshall, he said he eventually had to drink a broth of the bacteria to prove that it would cause gastritis, and all the gastroenterologists were against him for over 10 years. So it was just interesting. I could make it short, I guess, or I could skip this for another time.
Yeah, I'm trying to see how this fits our lab meeting. You can, I mean, since you made the slides, you could be very quick, because I think that, it's an interesting story, but I also think I don't know how it will, I mean, it's general science story, but yeah, let's just make it very quick, because I was
Sure,
thinking
and-
more of a journal club, because I think I sent you an article.
Oh, that's there too, but can I start off with this one because it's more lighter? And I think I could make it within 5 minutes. And also, if you feel like it's getting off the topic, you could always say, and then I will change it so it's more focused on, the lab meeting if it goes to general science. Understand?
Okay, let's do this, because we have limited time. Let's do this at the end. Let's do your article first, and do other people first, and let's come back to this.
Sure, that's great. So I already did part 1, so this is part 2. And so this was the paper that was put in Nature Medicine. That's very like a good journal. And
Yes,
it was
well, you see, I'm sorry, I just have a question. Who did I send the Lunit journal to? Lunit paper. Did I send to Eugene or Juhi? The Lunit JCO paper.
I think it's Juhi.
Juhi, are you presenting it today, or when are you presenting it?
I had it ready for last week, but we were all busy, so I never presented.
I couldn't hear you, Juhi.
Oh, I had it ready for like 2 weeks, but we were always like over.
Oh yeah, we didn't have time to. Okay.
Yeah, so I
Yeah,
never
we'll see if we
presented.
can do it today after Sungmi. Okay. I just wanted to know who has what to present. Okay, go ahead, Sungmi.
Okay, so the title of this is Comprehensive Molecular Characterization of Clinical Responses to PD-1 Inhibition in Metastatic Gastric Cancer. And it was published in 2018 when there was a surge of other journals talking about similar subjects. So in this journal, this is a review from the last time, so we could get updated because it's been 3 weeks. So it was a single phase center with 61 selected patients with metastatic gastric cancer, and they were all treated with pembrolizumab, and this targets PD-1. So it's an immunotherapy in terms that it's a antibody. So this is the antibody we're talking about over here. And then that could induce T cell killing of the tumor cell. So the goal is to identify the predictive biomarkers so that we could select the type of gastric cancers who will benefit most from this type of pembrolizumab monotherapy. And yeah, so how would they identify the predictive biomarkers? So with these 61 patients, they did tissue biopsy with these testing mechanisms to test like the subtype, and the subtype is detailed later on, and mutational load of the tumor. And they did RNA sequencing, and they see if a specific EBV virus that is known to interfere with DNA sequencing, and I'm not sure actually. Yeah, so to cause DNA mutation actually, they sequenced it if they were infected with this virus. And they also did what Dr. Che does in his clinic also, is plasma-derived ctDNA, which is DNA that is shed from the tumor or other cells in the body that is not part of the cell, it's just the DNA that you could find in the blood. So it's not invasive to obtain this liquid DNA of the tumor. And so, I put the conclusion here first at the bottom. So if you have a positive expression of PD-L1 patient, then it would work. The chemotherapy would work. And if you have microsatellite instability is high, it would work. And there's quite a difference here. And if you have a positivity for infection of Epstein-Barr virus, then the immunotherapies like to work. And the results is also something I did last time. So I just want to go through briefly. So basically, complete response, partial response, and stable disease. The complete response is the most favorable. Stable disease means there's no progression. And if you realize, then with a median follow-up of 16 months, the overall response rate was just 24, and including the stable disease, it was 57. And so, and if you look at something that's interesting is with PD-L1 is 50%, with macrosatellite instability is 85, and with Epstein-Barr virus infected people, that's 100%, and that was the more new interesting thing in this study. So now from now on is the second part, which I didn't mention in the earlier presentation, is this content. Let me see. So it's about the circulating tumor DNA actually. So circulating tumor DNA is sequencing. It predicts response to pembrolizumab, and they use Guardian, which Dr. Che also uses. And change in ctDNA immediately post-treatment predicts response to immunotherapy. Yeah, so that's the result, and that's how they presented it in the graph. And if I'd explain this in more detail, so as I mentioned earlier, for people who are not too familiar, ctDNA is circulating tumor DNA. It's less invasive. It's from plasma, basically, no need for biopsy, and it can measure tumor mutational load as well as specific tumor mutations that wouldn't be found in regular cells, regular DNA. And Guardian 360, it has commercially available gene sequencing panel. And then we got this from 23, these people got this from 23 patients, and there was a generally high correlation with tissue mutation load. ctDNA mutation load kind of correlates with it, as well as the fact that it also correlated well with clinical response to the pembrolizumab. And then this is how they presented the data. And if you see, the more the CT mutational load score is, the better the response the worse the response is. So progressive is, wait. Yeah, so, yep. And I'm just sorry, I'm just getting confused. And then if you see the proportion of ctDNA change and response, if you see the higher the proportion, then there's more chance of progressive disease. And then this is progress-free survival according to tissue mutational load versus tissue mutational ctDNA mutational load. And if you see in the graph, you can see that there is like a certain correlation with ctDNA, the progression-free survival falls really low. And it's also with tissue as well. Yep.
So stop right here.
Yep.
So can you make the graph bigger?
Yes.
Can you make that as full screen or something? Yeah.
Yeah.
All right. So keep in mind, we have a lot of stage 4 patients with gardens, serial gardens, and some serial Signatera on every 6 weeks. So because of, I guess because of the bandwidth, we weren't able to analyze stage 4 yet, but Sungmi has some data with her. And there are more and more patients who are getting Signatera for patients who are stage 4 or 3 advanced. And we have garden data too. So what we can do, oops, it's blocked, is do a similar approach. ctDNA mutational load. And the mutational load is like blood TMB. And according to that, this is gastric, and so high mutational load is likely to be MSI high. In lung cancer, that's not the case because there's no MSI high, almost none. But here, the higher the mutational load, the better the response, with only one exception. So that's something we can explore as well. And then graph C is according to the mutational load, is there a difference in PFS? So they did look at both here, which we are also interested in. Tissue, mutational, that's high TMB, ctDNA, high TMB. Those are in a darker line, and they both show better prognosis compared to the low TMB. I think this is really true for gastric, where MSI high is a really distinct entity. In lung cancer, it's not. So I'm not sure in lung cancer if we can see this kind of phenomena or not. But we can look at it. Sungmi has all the data. Now look at graph D, proportional ctDNA change. So look at the very first patient. After treatment, ctDNA has increased by more than 10-fold. What does that mean? It means it's simply not responding. Maybe hyperprogression. This is tragic to have tumor 10 times bigger. In terms of the ctDNA load. So we can look to see what is the fold change. Okay, if it's a 50% deduction, decrease, then there's likely to be a good response. So it's no brainer. However, actually to prove that statistics in the real world setting can get you in Nature Medicine. Okay, it's a very prestigious journal. They've shown this, and also they showed EBB relationship, which is very interesting for gastric. It's very unique. So the next question is there any virus that does similar tricks for other type of cancers outside gastric and gastric. That nasopharyngeal. EBV virus is endemic in South China. And if you got EBV, you're likely to get nasal cancer. If you have EBV, you're likely to get stomach cancer. I feel like there are more viruses responsible for our cancer. We just don't know yet. There's more discovery to be seen. But what's amazing here is that EBV predicts 100% response to immunotherapy. Why? Having EBV does not mean your MSI high, your PD-L1 positive, your TMB high. It doesn't mean that. But EBV is such a strong predictor. It's a virus. It's just a sign of viral infection. Okay. So what is with that? And can other histologies have such similar things going on? That's a golden question.
Can you make the argument that EBV causes these tumorigenic cells or these mutated cells to present more MHC and kind of
become more antigenic? Yes.
Yeah.
That's probably the mechanism. That's probably the mechanism. That same thing applies for HPV. That's and then there's Merkel cell carcinoma from skin, and they do the same thing. They have the same HPV virus. They don't have high TMB. You only need one oncogenic, one antigenic strong immune trigger. Anyway, so that's when things get tricky. If you have other confounders like that, the TMB doesn't become strong marker anymore, because you only need you don't need high TMB for good response. So it's heterogeneous entity. I'm telling you, these articles have great lessons inside. Anyway, look at graph E. Decreasing ctDNA that dynamic can predict good prognosis. So is that also true for lung cancer patients? And I also see a lot of gynecologic thyroid cancer patients. So is that true for other tumor types? Can we replicate the same graph, B, C, D, E, for the data that Seungmi has? So that's the next question. Seungmi has all the data. I think Seungmi, Juhee, Arnie, and Eugene can work on it at their leisure time. Okay. At their leisure time. I know there's priority, but I want to tell you that this is the very graph that we need for the next SITC, or next AACR conference. We wanted to look at it, but Seungmi did not have the bandwidth. We decided to look at the curative treatment patients. Okay. And we might find something like EBV positivity. Maybe there's something else that's correlating, because no one thought EBV would correlate this well. Can you go back to the EBV graph to me?
This one?
Yeah.
Yeah, I think so.
So here's the CR and PR.
This is not EBV one. I don't think I put the graph for the EBV one in here.
But any
Would you want
data,
me-
send me the data.
It's not in this slideshow, unfortunately. I only selected a few graphs. Do you want me to bring up the
Wait.
article really quick?
So is the EBV marked in this graph or not?
I think the green is the EBV. In green.
Green is EBV?
Yeah, because it says they respond regardless of mutational status. So
Okay.
the green is
Green is EBV positive. So all 5 EBV positive patients were PR. That's the point. It's 100% in their data set. I mean, it's small, but it's amazing. And for stats, for our Tempest NGS, I think most papers, there's an algorithm to say whether they have Appleback signature or HRD signature or MMR signature. So I think MMR is not really relevant for lung cancer, but at least HRD and Appleback, you can run that algorithm to say if this sample belongs to that Appleback signature positive or negative, So that's for your future manuscript. But anyway, no, I think it's a beautiful data. And I already gave you a set of graphs that we need for our data set. So Seungmi has all the data, and Seungmi, you can teach Eugene, Juhi, Arnie, Sha, everyone in the lab so they can carry on your legacy.
everybody's smiling at the lab. Yeah.
If you finish all your manuscripts before the April 8th, you can initiate this project, okay?
Yeah. But if I can't get anything done by April 30th, like I'm staying here until mid-May, but I'm just gonna hand over. If I can't done it by April 30th.
Yeah. No, what I'm saying, because you studied a lot about ctDNA. You are the guru of ctDNA in the lab. So I want you to teach your legacy to
I'll try my best.
in the lab so they can continue to do the project with the data set, okay? So I'm telling you, this would be our next project for our lab. And I don't expect you to finish this one, but it is something you can still continue to work and teach other lab members, okay? Okay, yeah, carry on.
Yeah, I'm done with my presentation. Oh, wait.
Yeah.
Oh, sorry, the discussion. So it's basically I didn't go because I thought this would be a short presentation. I didn't go into the detailed discussion, but it's basically a statement of the previous things, like which biomarkers that we could test, genomic biomarkers could be helpful in seeing which patients will respond best to pembrolizumab. And it's a restatement of the things I've mentioned. And I didn't go into too detail. And it does mention like one patient, kind of like Albert Curie, who had gastric cancer and liver metastasis. And he went curative resection following 12 cycles. So, and then he eventually achieved PT0 N0 following surgery, and is currently still receiving pembrolizumab. So his so after pembrolizumab, then he had the curative surgery, and he's still no cancer is found. And this guy was also interestingly EBV negative, microsatellite stable, but he had PDL1 positive. So EBV minus and MSS stable, microsatellite stable, they don't favor him. PDL1 positive does favor him, but he still responded really well. So these authors are saying there are always exceptions to biomarkers. And some people could still benefit from regardless. Yep.
Great, thank you. Was there a mesenchymal marker, or I think you skipped something?
Mesenchymal, yes. About the groups, that's mesenchymal is with TCGA. That's mesenchymal is a subtype of the gastric cancers that is very, I think it's not a good one.
Can you show
Unfortunately, I cut that part out because I will have to explain too much about the basics. Unfortunately, by now I've forgotten it. I'll have to go through. I'll need like 5 minutes to remind myself of the results because it's been like 2 weeks since I read it. Um,
But you don't have the PowerPoint
oh,
on?
yes.
I thought you did.
Um, no, I decided to cut it out. No, sorry, I think I cut it out. Oh, no, I do. Sorry. Sorry about being not aware of my own presentation. So molecular classification by gastric cancer, biology, this, blah, blah, blah. So they were known molecular subtypes. So there were two different groups: the Asian Cancer Research Group (ACGR) and TCGA. They have basically classified gastric cancer more than using molecular classification and gene expression data. It's more than just saying stomach cancer in the antrum, stomach cancer in the body. It's more than that. They're using EBV positivity, microstability positivity, and other factors to subdivide into usually 4 groups.
Yeah.
And so mesenchymal subtype, which would be shown as EMT positive, is a type that's in ACGR. And then if you look at the overall response rate, is 0% versus 30%. So
Yeah. So I think this is another big point you need to learn from the manuscript. So this is relevant for your stats because all the Tempest data, we have RNA sequencing, and there's now going to be some algorithm to say, is this a EMT phenotype or not, or mesenchymal phenotype? And gastric breast cancer, they have the most well-established molecular subtypes by RNA sequencing. So what I mean is, people will know, oh, this is mesenchymal type in gastric, or this is, this is a triple negative type in breast cancer. They will know, but in lung cancer, it's not established. It's not established. A group in Southwestern, Dr. D. Mina, Minna, you can write it down, and he has established like 7 molecular subtypes in lung cancer. He's been publishing in that field. So I would say establishing, like, run some like algorithm from our RNA sequencing from our own Tempest data and establish like some certain RNA expression based subgroups is what we need to do for our manuscript class. So that's something that we need to do for our lung cancer patients as well. So it's pretty powerful. If you're mesenchymal, not 0% response. So it's predictable. So I think AI or some sort of algorithmic score predictor should all put transcriptomic virus status, MSI high status, the tumor burden status, mutational load status, everything combined, LUNIT score, LUNIT, everything combined, radiomic score, and come up with the strongest predictor. I think we need to do this. And Stas, perhaps after you finish your step 1, I want you to do a journal club. So there's a paper that just came out from Houston Methodist Hospital, and their group have done a mathematical modeling of response to immunotherapy that I will copy and then put it in our lab messenger room, so you can present that paper when you have time. But I think we need to look into what's the best modeling of these predictors. What's the best scoring system? So I think we can start that conversation
Yeah,
Uh,
definitely. I think you did have me present a paper on a similar topic a few months ago.
Right, right.
I don't remember what the conclusions were, but I vaguely remember that there were one or two factors that were significantly more predictive than all the rest. I just
Right.
forget what those factors were. Maybe it was TMB and something else, but anyway. Yeah.
Yeah. So yeah, we need to, update ourselves with the most recent advances in the field. So good job, Seungmi. This is really
Thank you.
interesting. Thank you.
Oh, I do have one question.
Okay.
So I was wondering why, so like if you look at the extreme, like extreme result, like, like 100% response here, the CIN 5%, and then mesenchymal subtype, it was 0% versus 30% in non-mesenchymal subtype, which shows how bad of a prognosis you would have if you have this. But they also excluded MSI high and EBV gastric cancers from the analysis.
Mhm.
And, and then the overall response rate for mesenchymal subtype was 0% versus 10% in the non-mesenchymal subtype. So it was still present, but I was wondering why they would try excluding these guys from the analysis. Is it to avoid like what was it? Yeah, sorry.
Compounding effect, maybe,
Yeah.
maybe.
Mhm.
So I'm telling you, you can do whatever you want, and then people will, you just have to read the method of how, sometimes you don't agree, and they're, if I was the editor, like, why did you do it? Include them, and then they will have to include them. You know what I mean? It's almost like it's whatever you think clinically makes more sense, you can analyze it that way. And for us, so I think it makes sense because MSI high is such a strong predictor, EBV such a strong predictor, so that if I add that, it's going to skew the data. It might trump all the molecular signatures and really not really confuse the picture. So the strong predictors, I don't want to deal with it. I'll just take them out and then look at the rest. You can do that, and there's no rule. As a scientific community, as a clinical community, if a lot of people would think that makes sense, then you can do it.
Yeah, to see if there's like confounding or modifying effect. So yeah, that makes it just makes it more convincing, I guess, that they did this extra step as well. So okay, thank you for the answer.
You're welcome. Okay. So yeah, keep me updated on your progress on the manuscript and anything else you're doing. And also, Seungmi, as you write manuscript, work with Arnie, okay, to create those 4 graphs, okay? I'm not asking you to do it, but tell her how to make those graphs from this article, okay? From your stage 4 patients. Okay. So that, I mean, she's going to be in the lab for next 6 months. So I think she can do it. Okay. Okay. We'll go to Juhi to talk about the journal club article first and then her project next.
The journal club
Yeah.
presentation first.
Yeah.
Uh, uh, could you unshare? Oh, sorry. Oh, it's been a while since I made this. It's not very fresh on my mind anymore, but I'll try my best. I don't have a mic. I don't know how it's getting my voice, so you can mic. I don't think so. It has nothing. Is this the mic? This is the mic. Okay. Sorry.
I hear you okay.
Okay. Okay, so this is my presentation. So I basically read this journal like 2 or 3 weeks ago. Um, it's a it's entitled Artificial Intelligence Powered Spatial Analysis of Tumor Infiltrating Lymphocytes as Complementary Biomarker for Immune Checkpoint Inhibition and non-small cell lung cancer. So I just think it's nice to go over the abstract because it like summarizes everything. So if I briefly go over it, basically they're gonna talk about biomarkers on the basis of tumor infiltrating lymphocytes. Um, um, and that's gonna help them look at how effective immune checkpoint inhibitors are gonna be. And to do this, they came up with the artificial intelligence powered WSI whole slide images analyzer of TIL that will help them classify everything into three immune phenotypes: inflamed, immune excluded, and wait, I can't see. And immune desert. And this these three types we're correlating with the tumor response to ICI and survival in non-small cell lung cancer. So their results were that the inflamed one actually showed enrichment in local immune cytolytic activity. It had a higher response rate and prolonged progression-free survival compared to the other two. And at the whole slide image level, there was like a significant positive correlation between the tumor proportion score as determined by the model, so the artificial model, and the control TPS, which was the ones that were analyzed by pathologists. So 44% of tumors were inflamed, 37.1 was immune excluded, 18.9 was immune desert. And the incidence of inflamed in patients with programmed death ligand 1 TPS were these numbers respectively. And the median progression-free survival and overall survival were 4.1 months for inflamed, 24.8 for wait, I'm a little confused. Oh, progression-free survival was 4.1 months. Overall survival was 24.8 for inflamed, and then the others respectively. So their conclusion was that this artificial intelligence powered spatial analysis of TIL correlated with tumor response and progression-free survival of ICI in advanced NS non-small cell lung cancer. And this could like be used as a supplement biomarker, yeah, to help like predict patient outcome. So I'll just briefly go over everything, not super fresh in my mind. So currently the diagnostic test that they use as like a supplementary for immune checkpoint inhibitors in non-small cell lung cancer is PD-L1. So they were raising the question that they needed something new, a novel biomarker to predict this response in the clinical setting. With especially in this subgroup of patients that had met in non-small cell lung cancer with PD-L1 TPS 1 to 49% because they responded similarly to chemo and immunotherapy. So like it would be nice to narrow down who these persons were going to be. So and they suggested that TIL would be a promising biomarker, but it is very challenging to use it because it's hard to come up with a cutoff point, and it's also very it requires a lot of work. So because it requires someone to go through all the images. So what they did was they came up with this artificial intelligence powered analyzer called Scope IO, and that artificial intelligence analyzer was going to do the work for them, basically. Yeah. So this talks a little bit about Uniscope IO. It looks at the tissue region uh, from and from a whole slide image of 3,166 images of 25 cancer types. So, and these are like the databases they use. They use samples from um, the TCGA and SMC and SMUDH. So basically, this is the method. I'm just gonna skip over and then go straight to the results. But before I do that, I kind of want to explain a little bit about the three phenotypes. The inflamed is when the TIL density is more in the cancer epithelium level, but above the threshold, and the immune excluded is when the TIL density is in the epithelium area below the threshold or in the stroma above the threshold. And the immune desert is when the TIL density is in the epithelial area and stroma, but both are below the threshold. So it's a little confusing when you look at it. It's better if you look at the results and see actually the diagrams. It explains it better. So basically, these are the whole slide images if you see figure A. And then they put it into grids. And then they randomly select these grids, and then they look at the cancer epithelium, cancer stroma, and the TILs, like where they're in the grids that they are looking at. And then they do this AI model training first before making them analyze. And when they start analyzing, then they tell you like if it's inflamed or if it's immune excluded or if it's immune desert. So yeah, that's basically how it works. And this is figure 1C. It shows like figure 1C shows yeah, it basically shows the sample images that they were looking at, that the Lunetscope Bio was looking at. And then 1D shows the internal validation that they did. And for the epithelial, it was 0.97. For the stroma, it was 0.95, and CIL was 0.92. And then they also did like an external validation, and the values are like comparable. So yeah, they looked at how sensitive or how specific it was going to be. And then
So what is, so CE stands for what?
cancer, wait, let me go back. Epithelial, I wrote it down. Sorry. Oh, I never mentioned this. CE stands for cancer epithelium. Yeah.
Cancer epithelium?
Yeah.
Okay, so
And then
epithelium.
CS is the stroma. So cancer stroma.
Hmm.
Yeah. So as I said earlier, they took samples, they took images, and they made it into grids, and these are called samples, and they were classified into these three types. And the proportion of grids classified were this. So if you look at figure 2A, for the TCGA cohort, it was so the figure doesn't show how much it each was, but in the TCGA cohort, it was 24.8 for inflamed, 49.5 for immune excluded, and 25.6 for dessert. And then they go on to break it down for the other two cohorts too.
Can you go back?
Or this? Is that
Yeah.
what you were asking for?
Yeah, this one. So
Oh, this is the inflamed. So the upper two images show the inflamed, where you see more of the TIL and the epithelium level.
Mhm.
And then the next one is immune excluded, where you see TIL less in the epithelia and more in the stroma.
Yeah.
And then the last one is dessert, where you see less of them in both places.
Mhm,
And
mhm.
then they so this is like the grid level, and then they were like, and they took the whole image, and then they kind of showed how inflamed it was, or like how immune dessert or excluded it was, and then they like transformed that into a graph, and they called it the inflamed score or the immune excluded score.
Mm, mm, mm. Relative density and immune inflamed score and immune excluded score percentage. Interesting. And WSI means what?
Whole slide image.
Huh?
Whole slide image.
Whole slide image.
Yeah. Basically, when you cut up the pathology into like slides, like, that's what they were referring to as WSI. And then
Okay.
they further divided into grids before like putting them together back again.
Mm. So I guess the idea is in one whole slide, you'll have areas of
Yeah.
inflamed,
Yeah.
excluded, and then
Yeah.
desert,
Yeah.
and look at their percentage.
And then depending on their percentage, they're going to give them a score.
Yeah. So I think what's amazing is there's a lot of complexity here,
Yeah.
but then it boils down to a one number. What is the score? And how do you accurately have that one score reflect the what's happening?
Yeah.
And then still have that one score. So it's like it's called deconvolution.
Yeah.
How do you there's so many data, but you make that into a one number, and how does that one number predict so many people's different treatments and then treatment outcome? Like it's almost like going this way, funnel, and then you expand the indication. So
Yeah. So basically, the way they came up with the score is that if the inflamed was above 33.3%, then they call it inflamed.
yeah.
Yeah. And if the immune excluded was 33.3% or above, and still had an inflamed part two, then they put it as excluded, and then otherwise they put it as immune dessert. So yeah, it was kind of cool how they did it, how they actually made AI about.
Okay. Yeah, proceed.
Okay. So figure 2B shows the geometric mean value of GDMA, G, or FP9.
Yeah.
It shows this value to be significantly increased and inflamed IP, if you can see the red one right here, compared to the other two samples. And figure 2C shows that the tumor samples with high tumor mutation, mutational burden, so tumor mutational burden is TMB, it was 23.9 and 23.9 in inflamed, and 22.4 in immune excluded, and 15.1 in immune dessert. Yeah, so that's what they showed.
So go back to this slide. So no, the stars had to leave, but I guess one thing that that we could do, and Eugene O, you and Arnie, this may be relevant when you talk with Stas. This is something that we can ask Tempest, because what they do is they get slide from us and they scan H&E, So they have image
Oh.
of the slide. Okay,
Mhm.
so let's ask Tempest to see if they can send us the image files of the slides, which then we can take it to Lunit and get a Lunit score and Lunit
Oh.
type.
Okay.
And we have RNA sequencing where we can look at all these expression,
Mhm.
TMB, everything, and do the same correlation. So it will be it could serve as a validation paper. So this is coming from
For what?
Samsung Seoul Hospital,
Yeah.
coming from Bundang Seoul National University Hospital, and then we can check whether this same pattern holds true
Oh.
with
okay.
Northwestern data.
Right,
Okay,
right.
we have everything. So let's see if Tempest is willing to share H&E slide image files. Okay, let's remember that.
So this Lunit, it's like accessible to all researchers. Is that what?
Well, I am part of their advisory board, so I work with them very closely.
Mhm.
And
That
they,
would actually be really cool.
yeah, Easer, Easer, and Eugene O work together to send our own Northwestern patient slides
Mhm.
to Luna.
Lunit.
Yeah, and they are analyzing our data. We have a research agreement.
Okay.
Yeah, so we have a lot of fun things happening.
This could be your project.
Dr. Shay, can I ask one quick question? So
Uh-huh.
in terms of patients you want us to get to the slides, the
Uh-huh.
patient list would be the one that Stas showed us in this meeting, or do we have to collect the patient number or
No,
list?
no, no. So
Just
Stas
ask Tempest.
talked about 300
Right,
patients.
right. Okay.
For
About
that
the patients.
300, can we also
Okay.
get the image files?
Mhm. Got it.
That's the question. Yeah,
Okay. Thank you.
and you can CC me and just email the Tempest person.
Mhm.
And Dr. Che is asking if we could get access to the image
Images.
file of the H&E slides that you have from our patients, so that we
Okay.
can analyze the, you don't have to mention Lunit, but we can analyze
Okay.
the tills and everything. Yeah.
Got it. Thank you.
The other thing is it's not just Lunit. Tempest is doing the exact same thing Lunit's doing. They have their own metrics. So we can
Oh.
analyze their metrics if they want.
Okay. Then yeah, let's see what they respond.
Yeah.
Mhm. Got it.
Okay, go ahead, Jui.
Oh, yeah. So, then- so if you look at figure D.
Oh, well, go back. So this similar figure, we have it in our paper. So we published this in American Journal of Pathology. The lead author is Dr. Peter Cho. I'm the corresponding author. So this paper, Peter Cho and Dr. Chung-Han Park, they're the lead authors.
Mhm.
And it came from our lab. And we published all the similar findings in the American Journal of Pathology. It's accepted and the journal will be published in next few days
Wow.
online. So Peter just emailed me. I saw the email that it's fully accepted and it just will be published in any day. So very similar findings. So yeah, definitely read that paper once it comes out. Next.
Got it. I'll look at it. Yeah.
Okay, yeah. Go ahead.
So if you look at figure 2B, it shows, favor inflamed or favor immune excluded. That basically means by they did a gene set enrichment analysis, and it showed interferon gamma and IL-6-3 to be highly enriched and inflamed, whereas P53 pathway and WNT beta catenin signaling was more enriched and immune excluded. If you look at figure E, they show that in inflamed IP, the composition of immune cells was more like CD8 positive T cell, memory T cell, memory B cell, and more macrophages. Whereas in like immune excluded, it was more naive B cell or M0 macrophages, and in immune desert, it's more neutrophils and M2 macrophages. So they did the diagram to show that. The diagram E, F shows that inflamed showed an enrichment of CD3 expressing or CD8 expressing cells. Immune excluded showed FOXP3. Immune desert showed CD68 plus.
Mm.
Yeah, so they did like the whole thing about what kind of immune cells were present.
So what is the difference between E and F?
Oh, sorry, I should have mentioned it. It's a different cohort. So E is from the TCGA Luad and Loops dataset, and figure E is from the SMC dataset, so this is the SMC dataset. So it's like different datasets.
So E is TCGA.
TCGA.
What is F?
TCGA
I think F is from SMC cohort. Samsung Medical.
yeah, F is from Samsung. TCGA is the cancer genome atlas.
Mm.
So they initially started off with 3 cohorts.
Mm. Okay.
Yeah.
So maybe Chuhi, can you do me a favor? Can you send this article and this the figure 2 slide to Stas, so that when he works on the squamous adeno, and he has the immune cell phenotype analysis, so maybe he can utilize this type of graph.
Oh,
So
yeah. This
just
graph is really nice, actually.
yeah, it's just graphically much enticing. So just send it to him.
Okay, I can do that.
Yeah, thank you.
And figure G kind of like sums it up in like an image format. They colored it up too, but it's not super visible. So
Yeah,
basically figure E and F is like summed up in figure G.
yeah. So these will give us thoughts. And if you look at it,
Yeah.
there's a lot of ones that we already know, but there is this estrogen response pathway. Like, what is estrogen doing here? You know what I mean? It's doing something. I published a estrogen pathway, how that modulates immune response. So that's written by Michael O, who used to be our resident here in Northwestern. He's now UCLA Hemonc Fellow. So it's published. You guys can look it up. Michael O, Yongguang Che, and estrogen receptor, and cancer or something, and you can find his article that we wrote together. So, what is Wnt beta? What is cholesterol doing here? What is fatty acid doing here? What is coagulation doing here? And how does that modulate the immune response? So this is and then what are the evidence? Who looked this up? Like, what's the group that is looking into this question? So this is opening a lot of questions. So this is why studying other research articles will open up your eyes and look for great topics, look into niche, and so forth. But yeah, go ahead to the next.
Mhm. Yeah, so these were like the more important results. The next one was the next is table 1, which shows the patient characteristics. So they show it by cohort, and so 518 patients were eligible out of 552, and according to table 1, most of the patients received ICI as second or more than third line therapy. Or more patients received it as third line or beyond. And SNU cohort, so 42.5%, 45.2%. The distribution of PD-L1 TPS status was similar across. There was no correlation between IP and smoking status, which is kind of surprising. Liver mets and lung immune prognostic index score. There was some correlation between IP and pleural effusion and brain mets, according to this table, which is wrong. And then they go on to make figure 3A, where they show the distribution of inflamed, immune excluded, and immune deserted IP. So in the SMC cohort, it was 42.1 for inflamed, 37.8 for immune exclude, 20.1 for immune desert. For the SNUPH, it was 46.6, 36.1, 17.4 respectively. So they noticed that the incidence of inflamed IP correlated significantly with PD-L1 TPS above 50% status in both cohorts that they looked at, which is the Samsung and SND cohort. And if you look at figure 3B, so they said the median PFS, sorry, with ICI among inflamed was 4.1, which is significantly higher than the immune exclude and immune desert, which was 2.2 and 2.4 months respectively. And then they said there was like a improvement in the overall survival after ICI treatment and inflamed patients, which was seen to be 24.8 months compared to the other two, where it was 14 for immune exclude and 10.6 for immune desert.
So I want.
So basically, they were saying that the response rate to ICI was high in inflamed IP. Yeah, compared to the others, like significantly higher, as the graph shows.
Right.
So, and then figure 3C showed that if they use a first-line platinum-based chemotherapy, then the PFS and overall response rate was not significantly different across the 3 IPs. But median overall survival after chemotherapy was pretty different. So it was 36.2 months for inflamed, and it was in the 20s for the other two.
So this is meaningful.
Yeah.
So when you say you know, a Lunit scope score is a biomarker for immunotherapy, it has to work only for immunotherapy. So the graph C tells us that it is. Now, if you look at the graph the overall survival graph that it seems that flame has a better
Mm-hmm.
prognosis.
Yeah.
Does that mean it is a predictive factor for chemo? Yes and no. I mean, for overall survival, maybe yes.
Mm-hmm.
So this is the same thing. We talk about EGFR mutation.
Mm-hmm.
EGFR mutation, they live longer because there's definitely EGFR TKIs, tyrosine kinase inhibitors. However, they also respond better to chemotherapy. If that's their biology.
Mm-hmm.
Okay, so it's very similar. Now we have to think, why is that true? And why would inflamed group uh um do better? And because they received immunotherapy. They received immunotherapy at second or third line. Overall survival will always be affected by a different line of therapies you received, not just the chemo.
Mm-hmm.
It's not like they received chemo and nothing else. Then the graph of overall survival, I think, would be same as the PFS graph. Okay.
Yeah.
So I think the conclusion is, I think the effect of the overall survival is still there. So the question is this overall survival graph and the graph you showed me before with the immunotherapy similar or different? So this is different because they probably captured the day one to be the immunotherapy start date, whether it's second line or third line, versus the platinum, you got day one as the chemo start date. So they're different graphs. Okay.
Yeah.
Yeah. Okay. Proceed.
Yeah. And then they look into the IPs in relation to PD-L1 TPS status. So the figure 4A, it explores the relationship between IP and PD-L1 TPS status. And if you look at figure B, it shows the incidence of it shows the incidence. So the incidence of inflamed IP in patients with PD-L1 TPS less than 1% was 31.7. 1 to 49% was 42.5, and above 50% was 56.8. So what they were suggesting was that the PD-L1 status correlated well with the inflamed IP, and like the incidence of immune desert IP was more common if the TPS was lower than 1%. Yeah.
So they performed better
Yeah. Oh.
in 1 to 45. And it didn't,
Mm.
I guess it did not perform well when it's 0%. And not sure if that's a sample size problem or maybe it just doesn't work that well,
Mm.
right? It's, yeah. Okay.
Yeah. Figure C. So the median PFS for inflamed and non-inflamed where it's 4.4 to 2.1 months for PD-L1 TPS in the 1% to 49% subgroup, and it's 6.2 for inflamed, 3.2 for non-inflamed for the above 50% subgroup. And for the overall survival, there wasn't much of a difference between the IP subgroups. And if you look at figure D, and the PD-L1 TPS 1 to 49% subgroup, the 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 prediction of tumor response to ICI by IP is 0.76.
So graph D is just for PDL 1 to 49, right?
Mhm.
Yeah.
Yeah, because
Yeah.
that's the one they were interested in for the most part.
Yeah. That is clinically the most interesting group.
Yeah. So yeah, so that's it for
So let me give you a comment. Well, I guess,
their
yeah, that's kind of,
but
yeah, yeah.
so they ended it off saying that the AI powered spatial trial analyzer was actually capable of predicting clinical outcomes of IPI in patients with advanced non-small cell lung cancer, and it was able to correlate the three IPs with local immune cytolytic activity, and it showed that patients with inflamed IP correlated with higher CR and longer PFS. And then uh, the Lunit scope IP can process these twice simultaneously with the PDL1 IP, and uh, then they suggested that this could serve as a complementary biomarker in addition to PDL1 TPS for advanced non-small cell lung cancer, especially in the group that they were interested in, the 1 to 49% subgroup that could either respond to chemo or immunotherapy. So they suggest a future prospective study of the clinical application of this AI in treatment.
Yeah.
For
So yeah, this was published in JCO, and we were trying to be part of this group, but the Northwestern process took so long. we weren't able to get into this paper, but I think it's a great data. It's almost like too good to be true. Like, is this real? Like, I look, when I first saw the data, so good to be true. And it's a good news for patients that they have a better marker now.
Mhm.
The biggest tragedy is you believing that the therapy will work and you get it and it doesn't work. A waste of your health, time, money, everything. You're gonna die sooner because of that therapy. So wouldn't it be great if a physician will have a better biomarker to tell patients, yes, there's a very good chance this is gonna work. That's why I'm
Mhm.
recommending it to you.
Mm.
This marker came back negative, so this is likely not going to work for you. So I'm not going to recommend this to you, and move on to the next one. So this decision making is so important. It is life-saving step, and a better biomarker will is what we want, and that's why I invest my time, energy, effort into this. Um, so I do work with them closely,
Mm.
and this is the one of their biggest achievement in cancer immunotherapy world. So we're trying to open clinical trials using this Lunit scope in all cancers and also in lung cancer. So it's not just a paper. Oh, okay, it's a good marker. What's next? How the question is, how do we change guideline? Well, we need a
Well,
clinical trial. Okay, we can't change our clinical practice based on retrospective data. We need a prospective data, a clinical trial. And is this only relevant for lung? And of course not. This is developed pan tumor. And I think that's what's important. It's just not the lung cancer biology. It's important for all cancer. So think about it, how we can utilize this technology. Chui, oh yeah, apart from this, anything else?
Sorry, I have a question about this. So, but in order to have these images for the patients, wouldn't they have to have underwent, some sort of a surgical intervention? Like, is a biopsy like enough? Because it wouldn't be like of the whole tumor. So I was thinking,
So cytology cannot do it. A biopsy that has structure,
Yeah.
like the stroma epithelium, a biopsy of any needle biopsy is fine, as long as it contains a structure.
Mm. So would you say that like if there's like a tumor, the composition of, I guess, TIL across that whole tumor will be consistent? It would be either just in the epithelium or like mostly in the stroma, or like, you know, enough for a classification like this one? Or would you have to do like multiple biopsies for each person and like try to get an average?
Try to get what?
Like do multiple biopsies and like try to get an average in order to like come up with a score for a person, or would like a single biopsy be enough? Or like I wasn't sure how they got their like original data set, like,
It just came from a single biopsy.
oh, just a single biopsy.
Yeah. But if you have more than one slide, you can average. You can,
Yeah, I thought averages would be like,
yeah, you
more
can
accurate.
average whatever you have. But usually for diagnosis, they
Mhm.
make, I think, 1 or 2 slides. They don't make one. So whatever they have, you can work. You can still apply. But the more slides you have, the better and less bias.
Yeah.
So that there's inherent bias um, um, present in any tissue work. You know, what if you like biopsy here, not here? Are they
Yeah,
all
so
different?
that was my
They're gonna be.
concern.
We know they're all very different.
Yes.
So it's just by random chance that you'll get there, and that's their score.
Mm,
Same thing with liquid biopsy.
yeah.
You know, DNA fingerprints. You know, it depends on how much are they shedding. You know, and from different sites. But the better thing is that they're going to be a collection from all different metastatic sites all together in one blood.
Mhm.
So it's different than poking all the metastatic sites, right? So in that way, there's advantage
Mm,
of looking at DNA, but they don't have any structure. So for something like Lunit, you have to depend on tissue.
yeah.
That is, but tissue is not perfect. The people like to say tissue is the issue.
Funny.
But good.
Yeah.
So
So, oh, should I share about the other two?
yeah,
Do you want to share that tomorrow? Oh, okay, I can do that. So I'll do the case report thing first. So basically, um, we already did most of it, like organizing everything. So she went through all the progress notes, and then she like wrote down the how Vivian progressed over the time period we're looking at. And then Eugene was kind enough to make this really nice looking table that shows like the date, symptoms, workup, and disease or like treatment. Just so we can have like a nice picture of how the treatment is working or not working. And then basically the manuscript layout is what you told us yesterday. So the topic, my introduction. So Eugene will be starting to work on the introduction. We're hoping to get it done by this week. Well, at least the introduction and the body. So the body, I will be working with Arnie. Arnie will be helping me getting like images or the past results or like details, and I'll be writing it up. And next week, while we're gone at the conference, Arnie will research further on the other cancers that work with immunotherapy for the discussion. And she will try to start writing that up. So hopefully, in April, we'll finish it up as soon as possible for the case reporting.
okay, let's see what worked for her before. Let's go. I can review it. So the first systemic therapy is Lenvima. Did it work or not? Did it work?
The lymphema actually it didn't work, doc. So I make in the upper one, I make the comparisons of the imaging and the date of the therapy start. Up. I just make it, and so we can see from the lymphema it's not like decreasing. Stop that. Yeah, so we can see like from the start in May and
Okay.
in May, exgiva and lymphema, we can see like there's an increase size in number, no change of
Yeah.
left.
So that's not working. So that's why, like I told you, anaplastic is aggressive. Lymphema, a lot of times it doesn't work. So nivo-ipi didn't work.
Yeah. So I see there is like an increase even in metastasis until it around December, like November, or like around November and December, when you start change it into cabozantinib, there is like, and also you change it to temsirolimus, there is like some improvement.
Wait. So nivo-ipi alone worked.
Work. The
Yeah.
It worked.
Yes.
And then it didn't progress, but we added Lenvima, 10 milligrams, right?
Yeah.
And did it work? No, right?
No.
In
No.
large,
It didn't work. Lymphema didn't work in this patient.
but that was Lenvima added to nivo-ipi. Okay. It's not Lenvima alone. Nivo-ipi was never stopped. So you have to understand that. Okay. So here's what's tricky, and you don't have to write all this in the manuscript. This is for your own knowledge. Again, I told you how to write the manuscript.
Yeah.
You don't have to write the whole treatment outline. Now, here, what's tricky is that you switch therapy if there's a progression. There wasn't a progression, but I knew nivo-ipi would not work forever. So I wanted to add Lenvima to make sure that nivo-ipi continues to work. There are a plethora of data saying Lenvima can add increasing synergy between Lenvima and nivo-ipi. So I added it with that expectation, but I did not see any benefit. Rather, I saw progression. So that's why I switched to carbo, right? Added carbo in place of Lenvima, continuing nivo-ipi. So show me if cabo worked or not.
Carbo.
So after cabo, that was just the 10 days.
Oh.
And then the next, what is the next scan?
Actually, the way I see the cabo is started, actually there's an increased size and number of hepatic metastasis, even small pelvic ascites. Also in the MRI of the thoracolumbar spine, there is like a slight interval increase in size and enhancing nodules. I put it the bold one.
Yeah, so I guess this scan was done 10 days after cabo. And let's see, the next scan was done December 9th. Temsirolimus was started December 7th. So this December 9th showed some improvement. I'll go down. Worsening hepatic met still, right? So it's a mixed response, but there was, so that means cabo did not work. Now with the January scan, that it shows that it's working. So that's the effect of temsirolimus.
Oh, okay.
Okay.
I make
That's one of the December for cabo.
I make the line, that's why.
Temsirolimus is working. Everything is decreasing, and the most recent scan we got March showed that it's continuing to work.
There's no new nodule.
Okay.
Okay, so you got the whole idea?
Yeah.
Okay.
Yeah.
And what was the P10 mutation? Can you show me? Is there NGS or no?
What mutation?
P10. So you have to write down what mutation it is, okay?
Okay.
Okay.
No, I think I need it.
This one. The new one. Quadrant 360.
So any questions?
Not yet.
Okay,
I'll just start writing and then we'll have a lot of questions.
okay. Next,
And then there's the other one. So this is for the review, the systematic review of blood-based cancer. So we did like a preliminary read up, I guess, for background information.
yep.
And we kind of did a couple. We were organizing it, and then we saw we had enough background information to start looking into the actual articles. So
Uh-huh.
I did like a Pico because I think that's what they do for review journals, but I got a little confused. So I was a little confused about the comparison part and the outcome. So I understand that the participants of the journal population, I understand the intervention is going to be the screening. Comparison is like, would you say it's like the different essays, like the different screening essays, or
Oh, so comparison could be just very basic. This looks at proteins, this looks at methylation, this
so
looks
like
at,
the
yeah,
method employed
just the
by the essay?
method and the technical aspect. This requires 5 bottles of blood. This requires, 1 drop of blood. Like, just like really basics. How is the platform different? What is the requirement? What is the method? If it's a commercial, you can say, what is the price? You could say if it is this CAP certified, if it's certified or FDA approved, if is this anything, any aspect of essay you can imagine, you can compare between different platforms.
Okay, so some sort of aspects of essays.
Yeah. And how in a runtime, like does it take 7 days to run it, or like that's and any essay you could in the what was and then the outcome is you compare all the outcomes, like you wrote
Yeah.
down it in the below validity,
Yeah.
sensitivity, and reproducibility,
Yeah.
all that stuff. Yeah.
And then so I was looking through the databases, but like a lot of the articles were, well, not a lot. Like there were some articles from back then, like 1911 or something. I'm
Mhm.
pretty sure they're irrelevant, but I wanted to like put a date for the articles included, and I thought, like, would you be okay with like 20 years? Do you think that's enough, or should I include more years?
Hold on, let me call back my nurses. Let's do a 5-minute break. I gotta call back. Yeah.
Articles that are going to be included to 20 years.
Can you repeat that?
So the articles that are going to be included in their review, can I limit the date of publication to 20 years? So anything that's published on 2002 or after is going to be included.
Oh, that's fine.
Yeah, okay. Yeah, that was my concern. And so I did like a I did like a PubMed search where I got 3,400, a Cochrane 188, Embase 4,300. So Eugene removed the duplicates and we are down to around 7,000. I thought that it would be possible for us to like team up that way. That's gonna be the main team. So 1717 or 1718 articles per team. They're gonna assess it by title and abstract only, and then just put it down to like a lower number, hopefully. And then I will just randomly assign 1,000 articles for Sally to get a gist of what's happening, but I will not be using her input to decide on the articles being narrowed down for now because contacting her was a little difficult. So I'll put her more in the next one where we are actually going to start reading the full text articles and making a figure from the full text articles. And that's going to be the team.
Can you go back to the keyword?
So basically what we did was cancer and liquid biopsy or blood-based and screening or detection. Do you want me to
Screening.
add more?
Screening or detection?
For that, Embase took detection as cancer diagnosis. They like changed it up to that. So,
Okay.
so I was thinking, should I include early? But then I just didn't because you never know if they use the word early or not in the article. So. Did you want me to add more words to this?
Cancer detection. Early. What can be used in place of early?
I was also thinking like early or minimal or like some kind of sports like that. Cause like after a relapse, you would think of like a minimal or like lingering. But I wasn't sure. Like, I felt like there would be journals that don't use those words. That's gonna be like taken out of insert. So I just made it super broad.
Yeah. See what happens. I mean, it's too many articles, and we know there's only a handful of, papers out there, maybe less than 20.
True.
Should I do something like that? Early or minimal or lingering? Or
I don't think minimal or lingering is a good.
early
Early is good.
or pre?
Pre, but it has to be pre something. Blood liquid. I mean, it doesn't have to be. You can do blood dash based, but blood based or any blood. Liquid. Okay.
Actually, we tried blood only, but it came out with so many results. So we tried
That was like
blood-based.
in the 10,000. Like 90,000.
So look at what the Hopkins group, the cancer seek data, and then the gallery tests those publications. What are they? What words did they use?
Those were the articles we started reading and then got the words out of, but we can do it again. Like we can get more words out. That could work.
Early detection,
Multi-cancer.
early cancer detection, blood test, sensitivity of multi-cancer detection, methylation. So in the methylation, there's no word blood or liquid, right? So
Mm,
it just says cell-free DNA, the last one.
Oh yeah, maybe we should include that cell-free DNA or plasma.
Yeah, or plasma is a good word. Yeah, plasma or serum. You can even put serum because in the serum you get protein. So
mm,
blood, serum, plasma, liquid, or cell-free DNA. But you're gonna miss if they look at, let's say, exosome. They are in the blood. um,
mm.
can't be perfect, but so those are one category, or, and then as a category end.
Yeah.
And some people put early, some people don't put early, so I don't think it has to be end. Yeah, so look at other papers and see what criteria can include all the recent publications that you know of, and go from there. Okay.
Okay. We'll go through that. Yeah. So once that's decided, then we will work on 4 groups. I don't know if Shah wants in. We'll ask her when she comes here. If she wants in, then it's gonna be 4 groups working on the.
I'll just add her.
Oh, okay. Okay. So it'll be like 4 groups. Working on the journals by title and abstract, and then we will narrow it down to a certain number, and then I put Sally in the groups too, where we're gonna start assessing it by full text.
Yeah.
Like the figure 1. Figure 1.
Also reach out to what's her name? Oh, never mind. I think she's writing the variant project, so never mind. Yeah, just work with Sally.
Yeah. But I'll put her in the second process, not the first one.
Oh, the other one is Dean. Ask Dean. Dean was on our lab meeting.
Oh, okay.
Yeah, ask him. I think he'll be a great guy to be involved. Yeah.
Okay.
Just send him a message.
Yeah, I'll just test out the groups and reassign according if necessary.
Okay.
Yeah. Yeah, so after doing the full text, I want like the basic information. I'll redo it, but like this is what I want table 1 to kind of look like. Just like going through the basic stuff. And then
Sure.
we'll do more tables later on.
Okay.
Yep. So yeah, that's all I have for you. I'll redo the search. So
Okay.
yeah, I'll get that done today.
Okay. Sounds good.
Yeah, that's it.
So Eugene O, regardless of this, we need to set up Zoom meetings with the companies that are out there. I think we're just
Okay.
losing
For okay. Company for detecting CTCs or
yeah, CTCs or ctDNA. Either we need one for each.
oh, okay.
But I think we can talk to them because I don't know, who are interested in collaboration. Some might be we don't collaborate. You just order our test. That's all we care, and that they just want to make money. Some companies are really into doing collaboration research. So unless we actually do a Zoom meeting, we can't tell.
Okay.
Okay, thank you, Jui and Eugene. You can update us.
Okay. Can you unshare the screen? Oh, sorry. No, it's okay. So for me, I just want to touch base on the questions that Dr. Che mentioned while I was presenting the P2P person-to-person transmission of via cancer transmission via blood transfusion.
Yep.
So regarding the question about how did they prove these identical tumors between the donor and recipients, I look up and make a table.
Yeah.
So if you look at, so actually there were several, actually the article that I cited here, Strauss et al., this was not a case report. It was a review paper on the cases that have reported about the cancer transmission via blood,
Mhm,
transplantation. No, I'm sorry, it's not a blood, but transplantation. So
mhm,
I looked at each individual papers,
mhm.
and what they used, the methods were quite varying. So like some paper used HLA typing
Oh, good.
to,
That's
yeah,
strong.
and some paper used PCR based MS marker assay, microsatellite marker assay,
Hmm. That's good.
and some used some, for some paper it's just that histological examination, but they reported some markers like Pigmentin, SPEC, positive for melanoma,
Okay.
and they also said that they did tissue typing laboratory and fish analysis. So actually it was quite interesting. Um, so they found the XX chromosome on the patients, male patients, so they could notice that it's coming from the female donor.
Yeah, that's smart. That's smart.
Yeah, yeah, I agree. And for some, so because of most cases have dealt with melanoma, they mostly looked up for SPEC marker, and some paper also did PCR-based DNA analysis for polymorphic short tandem repeats. So they actually had this pretty good looking data. So they actually compared all the tissues from recipients and the donors, comparing all the letters here. And yeah, so and some paper look up the DNA typing of VNTR, I mean variable number of tandem repeats and short tandem repeats as well. So like you expected, like you anticipated, Dr. Shea, and most paper like prove it with genetic analysis.
Yes. That's great. That's great.
And yes, and then for other, so I also looked at the animal-based study, Batseli et al. This study was published in the Nature Biotechnology, and like unlike the human study, they actually controlled everything. So they injected actual circulating tumor cells from the patients into the mouse, right? So what they did was more pinpointing. So they actually looked at they run the facts, I mean, analyzing the cell by cell,
Mhm.
in terms of their cell markers. So they found out that so I'm not gonna like present this article because I have not studied it in depth yet, but the key point was that actually they discriminate even further, like metastasis initiating cells, so-called MICs among CTCs.
Uh-huh.
And the way they defined or found about the cells is that they looked up at the cell markers. So they defined MIC as cells expressing EPCAM, EPCAM, and CD44, CD47, and MET. So they looked at the specific MICs from the mouse to which they injected circulating tumor cells from metastatic breast cancer patients.
Mhm.
And yeah, so they also looked at the cell markers, not only by FACS, but also with this kind of immunohistochemical staining as well.
Mhm.
So these are the main methods they use to prove that that cells from human cancer patients did something in mouse.
So this is the immunochemistry of a tumor growing inside the mouse.
Right. So they injected the CTCs in mouses, like variant bones, I think.
Bone, okay.
Yeah, and then they detected like bone growth. Like they actually highlighted some osteolytic lesions caused
Mhm.
by bone metastasis,
Mhm.
and they analyzed the tissue with FACS and then immunohistochemical staining.
And that was the same tumor that was injected.
Right. So they compared it with all the cell biomarkers, cell surface markers,
Hmm.
CD44, 47, and MET, such things.
Hmm.
Yeah, so for protocol perspective, I think so like you mentioned, we need to reach out to the company, but I think it's better for us to prepare some materials to persuade them. So because we've known so many methods that other people used to prove some evidence so far, so maybe we can set up our protocol like at least in a rough way, like so
Agreed. Agreed.
can you give me any input? Like I'm gonna work on it if you can just give me some directions. So maybe and regarding that, like transfusion, maybe we can moving on to the transfusion review. So like you told me, I'm working on this kind of meta-analysis for studies delving into the association between the transfusion and the incidence of cancer.
Yes.
Actually, what I found was that there were more than 4 studies. I mean, there were pretty a lot. So I searched it again.
Oh,
So if
really?
you,
More.
yeah, I mean, and actually there is like even meta-analysis on association between the blood transfusion and then non-Hodgkin lymphoma. Like there was a meta-analysis already. They
For
proved it.
non-Hodgkin's lymphoma. Wow.
Yeah. If you
What
like,
was the result?
the result, yeah, if you see this, so this is the paper.
Oh, I cannot see it. You have to screen share again.
Okay,
It's blocking.
let me screen again. I'll share again. So if you see this, oh, sorry.
It's
It's a meta-analysis of observational studies, and it studied association between.
Eugene, can you I just want to finish that school of thought on the prior paper you showed. So in that mice model, the blood that was injected to the mice,
yeah, the cells, the patients.
they were just the patient's blood, right?
Right.
They were just circulating. So I think from that animal experiment, I think, the proof of evidence, what was the concentration or like how many, CTCs were actually injected, like,
True, I can share that.
yeah, so that information plus any other evidence out there that, hey, to have CTCs, grow in someone else's body, this is the density, this is the amount of CTCs required. If there's anyone who did research on that, we need to collect and have our own, because it has that evidence has to be reflected in our protocol. So we're going to talk to the CTC company. Hey, at least we need to get this amount of blood to be able to implant and grow in the recipient's body, right? So
Right. So give you just a
uh-huh,
ballpark number
uh-huh,
regarding that mouse study. So if you see the screen, so they collected 7.5 milliliter of blood,
uh-huh,
and they counted the number of CTCs in that amount of blood.
uh-huh.
And then actually the number was quite varying. You can see here, like, let me,
Oh, that's by each different patient.
right.
Okay.
So if you see this chart C, maybe table C,
Uh-huh.
and it's all draw from 7.5 milliliter of blood.
Uh-huh.
And there were actually CTC's number was from 0 to 4, and then it's up to 17.
Wow.
Yeah, a lot. But although the patient was number 1, just the number of patient was so small.
Yeah.
I think she might have so progressed. This is advanced disease, but yeah. So
A
and they
lot of
found out that, so if you see the frequency of successful grafts here in the right column, you see, so under 1,000 cell CTCs per mouse, there was no occurrence of successful grafts. I mean, no
Mhm.
tumor grow.
So yeah. So based on this, I think threshold is going to be around 1,000. So you could argue 1,000 divided by 7.5 ml tells us what CTC per ml is needed to engraft.
Right.
Yeah, so this is a really nice figure, and they used the company Cell Search company, right? So and that's
Right.
the company that is the one in Italy.
Yeah.
Yeah, so we should definitely do a Zoom call with them.
I'll contact this company.
Yeah. Yeah, and Leslie can help set up the Zoom meeting too.
Yeah, but
but
Dr. Che, yeah.
Mhm.
Yeah, like I have a question. Wouldn't it be an ethical issue that if we inject like some CTC containing blood to human, like wouldn't it be an ethical issue for us to allow that happen? I mean, if so, I feel like if I detect that amount of CTCs in the blood, I feel like I would not have give that to someone who need that blood. Maybe we should, yeah. I'm not so sure about the study itself.
That's a great question. So the original idea was to let me see.
So actually what we can do maybe is that we cannot operate any numbers about statistics, but maybe we can screen the potential blood that might have CTCs in that was supposed to be given other anyway. And then we can just follow up the patients. But still, I might feel guilty when that patient developed cancer from receiving that blood.
So here's the thing. This is an animal study. This is a cutoff coming from animal. Now the next question is, so I think we should get, so next question is, in the blood pack red bloods, that pack.
True. In the pack
And
RBC?
Yeah, that's about, I don't know, 200 cc's of
Maybe, yeah.
something like that. And so in that, if in the early stage cancer patients, what is the likelihood, what is the amount of CT that can potentially be in there?
True. Yeah, that would be a nice question to answer.
Because we can get 7.5 out of it. From it's still packed, but it's out of it, and run it as as a more like a screening project, and see, hey, in the real world in Red Cross, like what does each RBC pack contain? Can I find CTC? I might not find anything, right? These
Mhm.
are coming from actual cancer patients. The table.
Mhm.
We can do that. And if we cannot find any, doesn't mean there's no cancer cells in there. It's just that
It was
but
not detected.
yeah, yeah, but you could argue that if a 7.5 from 200 did find 0,
Mhm.
then probably there's not enough CTCs in there
Right, to be
to cause
detected.
anything. So maybe we will, if it's 0 CTC, we will, take them out from
Mhm.
our research.
Mhm.
If we want CTC detected
Mhm.
to a certain number,
Mhm.
then you could argue that they are included, and then we will track.
Yeah, it's warranted to monitor.
But if it's like 10,000 in there, then we're ethically worried, right? Is this
Right.
something? And
Very worrisome.
that's a
Yeah.
valid concern. So I would say the better, because we know who's gonna get it, right? But then we don't have a strong evidence to say that CTC
Right, that's
will
true.
actually cause
Will
cancer or
yeah,
not.
right.
And is this animal immune competent or immune deficient mice, or do you know?
Oh, that's a good question. So
Because good
mice.
immune system can clear this too.
Right. True. I think so. When I looked up the study, like roughly, I didn't see any comments on that, but
So yeah, maybe look it up and let me know
right.
later.
Right.
Okay, so that plus, I mean, I think the ideal one of other ideal scenario is to bank it.
Mhm.
We get 7.5 from each red blood cell.
Mhm. Already stored RBC.
Yeah, and then we do that for, let's say, first 100 patients. And once we get everybody, we
Mhm.
will run as one batch. So after several months.
Okay,
So we
after
don't know
several.
the data at the time patient gets blood
Right, the
transfusion.
blood. Right.
So, and then we just follow them up and correlate the numbers later, CTC
Mhm.
numbers later. That might be okay because right now there's no data that the human CTCs can cause cancer.
Right, true. There's
Plus,
no data like that.
and then we don't have that CTC number at the time of transfusion.
Right.
But I would,
Oh, actually.
because it will be run after we collect, let's 100 patients blood, we will run that later as a one batch.
True.
So
We
the
can.
question is, to do that, it has to be frozen, right?
Mm-hmm.
So do the cell search work
Mhm.
with a frozen sample, frozen and thawed, or would that kill CTCs?
Okay.
So
Oh,
that's,
the thesis can be preserved during that frozen
yeah.
period.
So that's a good question for the company cell search.
Okay.
Okay, so let's do a rough protocol and then reach out to cell search and other companies, and also gallery. We have a Zoom meeting coming.
True. I think it's gonna be tomorrow. It will be tomorrow. It's going to be tomorrow. So maybe I should
Prepare some rough draft.
prepare some rough draft by today.
Yeah, so I think the best scenarios is we get blood directly from the donor of transfusion.
Mm-hmm.
But that's too much.
If that's too much, we can do it
work with the
already
with the-
stored.
Yeah.
Got it. So yeah, so interestingly, like for non-Hodgkin lymphoma, there is pretty clear evidence that blood transfusion is associated with incidence of non-Hodgkin lymphoma. It was there.
In
So
general,
my question is, yeah,
it in general population,
yeah, they
high risk
although
population, or
no, they screened. So almost all studies were done in observational way, but they didn't restrict their population as some specific group. They just screened. So what they did was
I can't see your
retrospectively.
face.
Okay, I'll share it again.
Eugene, I can't see your face.
Yeah, right. Yeah. Yeah. So for these observational studies, so what previous researchers did, I can put it in this way. So one way is retrospective way. So they compare the incidence of compare the history of transfusion, getting transfusion between two groups. I mean, the one who developed non-Hodgkin lymphoma and the other control groups, and they compare the history of blood transfusion. So that's the one way they conducted their studies. So they didn't restrict it into some high-risk patients, high-risk group. So and so actually I found, yeah, as I said before.
So one
yeah.
thing you can do is you can look into the studies that they pulled.
Right. True. Actually, they included the Sweden and Danish group study as well.
Oh, good.
Because they subgrouped it, each cancer types, and they had the data of non-Hodgkin lymphoma.
Okay.
So I think they used it. So I think in terms of meta-analysis, the most studies I will collect could be divided into two, like the general cancer and the non-Hodgkin lymphoma. So do you think it will work, like to combine all the data together, or do I need to do it? I mean, already we have data for non-Hodgkin lymphoma, but what I'm trying to do is just see it generally, like without any discrimination of cancer types. So
yeah, I think you can do just all cancer, including lymphomas. And then you can.
Okay.
I think lymphoma. I think leukemia is a big one. Leukemia, lymphoma, and solid tumor are three big categories.
Okay, leukemia, lymphoma, solid tumor.
Yeah, but for solid, if you can still break it down like we did in our Korean data, lung cancer, other GI tumor, like if you can still break it down to subtypes, that would be good.
So just briefly showing the original Swedish and Danish data, I think it's pretty interesting to see. So if you see this, they actually thoroughly followed up all the nationwide data. So if you see this, they divided the follow period into some months. So from 1 to 5 months to 2 years and 2 to 4 years and so on.
Yeah.
And if you tracking down all the data, interestingly, the results are varied by the type of cancer. And even though other studies are few, but I think I can see the definite common results. Like, for example, liver cancer. So liver cancer is one of the most, where is liver? Yeah, liver, if you see this, over time, the risk of liver cancer is not that decreasing. I mean, it's still there.
Yeah.
The risk is still there. And it happened in other studies as well. Like, so yeah, it's interesting. And do you want me to like dissect all the cancer types and then see the incidence wise results, or do you want me? And so my suggestion would be like this review that we are working on. I think it would be to summarize in general, but I think seeing each specific type can be one another topics that we could make into another project. So
Yeah. Yeah. So if there's an interesting tumor type,
mhm,
because there may be a different mechanism, they are transmitting HCV, HBV, and that
right,
can work around and continue to cause cancer.
mhm,
So every cancer type might be a little bit different. Was there,
right,
so I think you should just, first of all, you should look at all cancer. And if you
mhm.
divide into leukemia, lymphoma, and
So
uh,
other like solid,
other uh other types,
yeah,
the common types.
right,
But whatever interesting observation you run into, just let me know and we'll see what we can. And then this could be a several different projects along the way.
right.
But
Yeah,
I
maybe,
for
yeah.
now, I think
Mhm,
we need a strong meta paper that will say, hey, blood transfusion really does increase
mm,
all tumor type, and this is a global phenomenon, and this is
mhm,
of great public health implication.
mhm.
And there is a animal model paper that CTCs from human can cause cancer. So now it's the prime time to really screen all our blood and show that this is really happening through tumor contamination.
Mhm.
Okay.
Right.
Because that's, we have different theories. Obviously, immune theory is relevant, and I think it plays a role. But what's really scary is the thought, and the immune theory is immune theory. I mean, if it's gonna happen, it's gonna happen. There's not many things we can do about it. We can do irradiation. We can do irradiation to kill donors, in lymphocytes, if that's really the story. But irradiating all the blood may, you know, I think it's just a cost issue too. But just the thought of having cancer cells go directly from donor to recipient is scary enough. And to show that
True.
that actually happens in human beings
Mhm.
is the whole point of our research.
Right. So I was very surprised that there were pretty a lot studies than I expected. So if you see the title, like in Japan, like they try to study the transfusion history and the thyroid cancer incidence among females, and like there are pretty several studies other than 4 that I previously like
Wow.
summarized.
This is
Yeah. So
amazing.
I think this can be another study subject for us. This
Yeah.
is so far I found, and then I have more things to review. So yeah.
No, this is perfect. This is perfect.
So so US in it was done in only elderly patient, and
Uh-huh.
so many studies were done for non-Hodgkin lymphoma. As you see, there's already meta-analysis on that.
Mhm.
And other than that, like yeah, it was from the UK, I think.
Mhm.
And yeah, it was from South Korea. And it's also done in,
So that's our paper.
yeah, here, it's our paper. Yeah.
Yeah.
Yeah, so I thought it would be done pretty easily because I thought there were only 4 studies, but like searching for it, I found more. So yeah.
Yeah, I think if you do a systematic search like Juhi is doing, you
Right.
probably
Actually,
will
that's
find,
what I'm doing.
yeah,
Right
I think
now.
you
Yeah.
can find more candidates. And I think this has to, it's like the Seokju Cho's meta paper. I think you should work on this as a, like a blood transfusion in
and then
general,
incidence of
more like,
cancer.
yeah, because he looked at more cancer patients, right? Now this is more like a general population meta-analysis, and I think it's a strong indirect evidence alongside our project.
True.
Yeah, so yeah, I think this is great.
Yeah,
This is great.
maybe I can involve many other lab members who are interested in this as
Yeah.
well. Yeah.
So definitely, you know, you could work with Juhi and
And
Arnie.
then
And
yeah, true.
anybody
And then
else
Sungmi as
interested.
well if
Yeah,
she's
Sumi, if
interested.
she's
Yeah.
interested. And
So yeah.
Sally and anybody who's interested, you can assign
Mhm.
them, you know, divide and conquer on this one as well.
I see. And for the table that I'm gonna put in the transcription review, so do you think it would be okay to make it like this? So the title will be summary of published studies on the association between blood transfusion and incidence of cancer. And then I'm gonna put the study and then database they used and the number of population they look into and then age and then key findings.
Yeah.
I know it's gonna be hard to put every detail in this table, but I'll try
Yeah.
my best to like, yeah.
So
keep
this
it-
is part of the review. So
Right. It's not gonna be in the meta-analysis, I think. Yeah.
yeah, so I think you should put key papers with the large number here. Select
Oh,
it.
okay. Mhm,
You,
selected.
to put everything here is too much. I think
Yeah, I think so.
you
Yeah.
selected and then just say you included the ones with the large numbers.
Mhm,
And just describe the pattern here.
mhm.
And then I think you need a separate meta-analysis paper, you know, systematically doing a meta-analysis on this topic.
Right. I agree.
Do you agree? Yeah, because it's too many papers. If it was just 4, that I think it was okay, but it's more than 4,
Yeah.
and maybe
True.
more.
Okay, I'll try my best to include some selected studies here. And
Yeah.
if I have questions, I will ask you. And yeah, other than that, yeah, I think I have to work on the raw transfusion study protocol from now on. And then, yeah, Albert Curie's case report, I'm working on it with Isol.
Huh?
And yeah, I'm sorry for delaying it. And I think I can show you the draft by next lab meeting, I think. Yeah.
Okay, good.
And do you think the title is okay with this? This is the title that we used in the abstract. But or I can just leave it like this and then ask your opinion next time.
I think it's okay.
Okay. And then for introduction, like I selected 2 key topics as IMA because it's what Curie had.
Yeah.
And then bilateral orthoptic lung transplantation. Do you think it will be
Yeah.
okay? Okay.
Well, you don't have to have a subheading, but
Oh, okay. Yeah.
for introduction.
Yeah. Okay.
Yeah, for case report, it's okay.
Okay. And for discussion part, I try to work on it first and then ask your opinion.
Okay.
Okay.
Yeah, for this, you can also, yeah, work with Juhi as well.
Oh, okay, got it.
Yeah.
Yeah, I'll involve her. Yeah, that's pretty much about me. And
Good. Yeah, let's get the case report out. I mean, there's a lot of projects.
yeah.
But
Yeah, I should
remember,
wrap
you
up.
guys are doing full-time research here, full-time, and it might be once in a lifetime experience. Okay, if you're going to be clinician, my life is always interrupted by clinical duties, all that. And look at me, I'm still trying to do these things, but I'm not full time. I'm
Hard time.
okay. So yeah, I'm very pleased by all your progress that you've shown us then. And yeah, I look forward to the next week meeting.
Thank you.
Anything else before we wrap up?
Mm.
So once we finish the editing of the 3 videos we generated for pacemakers,
Mhm.
I think we'll go on to the next set of patients. So work
Mhm.
with Liam to set up time for that shooting. I think we need let's say we have 3 teams. I want 1 video interview for at least once a month. That way 3 teams can generate 3. And maybe Seungmi, Juhee, Eugene can help shooting pictures of the hospital. So when you guys shadow me, I think
Mhm.
that was what the editor of Turano
Mhm.
publisher. So if you have nice, if you can think about a picture, you know, 3 of you can shoot some nice photos and send it to her together. We'll pick and choose the better ones. So we have 3 patients lined up, right? Songyeon Park, Marisa Wilderness. Who's the other one?
Oh, Louise Harris.
Oh
Not
yeah,
her.
better. She's better, but she's not
Yeah.
discharged. So
Oh,
if
okay.
she gets discharged home,
Uh-huh.
your next candidate. So follow her.
Okay.
Okay. Let's do that and we'll go from there. Oh, for I don't know. I think you guys can still interview too. Maybe we can interview Eugene O. You can share your story about that patient who is actually, doing the humor beats cancer or something.
Cancer. Yeah.
So you
Yeah.
share that story through our interview too.
Oh, yeah. I think I can if I can prepare it. Yeah.
Yeah, because that's a nice, heartwarming story. It
Yeah.
is a heartwarming story. Um, yeah, it is a heartwarming story.
Even though I was nothing but just observer, like just
Yeah,
I was even not even her caregiver, but yeah.
right. Yeah. I mean, I'm just brainstorming, or
Okay.
or anything that happened in 요양병원. Okay, in the
Mm-hmm. Mm-hmm.
any stories that are heartwarming that you want to share. Um, you guys who are here can still do an interview. You guys can
Okay.
be the one who shares those stories. So yeah, and then also there's Vivian's friend. Did you guys reach out?
Mm-hmm. No, actually, she was supposed to an email if she get approved from that patient.
Oh,
I
okay.
haven't heard about it, but I will ask her. I can ask her.
Okay. And we also need to get her consent, right?
Right, right.
Okay. Okay. Yeah, I think that's pretty much it. All right, guys. Let's talk more next week.
Okay, thank you for guiding us.
Bye.
Thank you, everybody. Bye.
Thank you.